circulates.
Yeah.
And some
Yeah.
other stuff.
Yeah. Yeah, a lot of
Took a picture and I posted it.
Yeah, we also see in between a lot of serpulates, so worms that are building tubes around, hard tubes, calcareous tubes, I think. Yeah.
So we found, if you were, you were there watching it, I think. Small, large, we thought it was sponges, orange ones.
Yeah.
And they are apparently solenogasters, which are like a group of mollusks. Very special.
Okay.
We
Ooh.
have to
Oh.
really
Just
preserve
to have 5 minutes.
Oh,
Yeah, let's
interesting.
go.
Oh.
There's still 5 minutes, guys.
Yeah.
Yeah.
Did you dish your jacket on the floor?
Yeah, don't worry.
Is that an anemone that is retracted?
Yes.
When has it?
No, I don't have some.
Do you want to have
Doesn't
fun?
smell like.
Yeah.
Let me toilet in.
Okay, no, I'm fine. Man.
Yeah, there you
Just
go.
if you want.
No,
No, okay.
Do you want me to jump in for a swap?
If you want, I'm okay. But if you want to,
If you're on time out, so you jump in if you want.
Okay.
so umbilical. Is it okay?
Yeah.
This one of these that has the arm, and I was just wondering whether you have any, or you need this arm to grab it.
We've also seen really cool solutions going on. Gonata maybe. And there are lots of shrimp everywhere. Oh yeah, now you can see them. They're all in the water column, and probably, yeah, they find a lot of food here.
Yeah, it will be. The hill will be on our right side, but if you want to follow the line,
Maybe a little bit of blue.
And we're talking a bit about the next directions for continuing
should be roughly
the ascent here on the summit of, or at least plateau of the seamount.
It's quite nice because
And then we have more lines.
Yeah, more or less.
So we're swimming through all the shrimp and little marine snow organic matter that we see here.
yeah, that's one.
And another beautiful rock wall coming up with many anemones and geodias, the
Yeah.
potato sponges.
Again, the amphidiscola.
And
The
the-
Hexactinelli with the stem, Procellides,
The worms in between.
plenty of geodias. I'm amazed how many geodias are here in this world.
Right, and then this doesn't seem quite white, so it's not working.
So we may answer these kind of questions: why here there are geodias and why geodias are not in other places, and in other places we have plenty of Hexactinellids. This you can try to explain, checking the environmental factors that we are gathering all the info from with the CTD, which is a device that is in the ROV, constantly collecting the temperature, the salinity, conductivity,
Oxygen,
oxygen.
Right now it's, for example, minus 0.5 degrees cold here.
Quite cold.
But mostly it has been minus 0.6. So slightly warmer. Even though we're much further north than during the previous dives.
It's a big one over there.
Big carnivores, or you mean the one behind?
The Hexactinellids.
The Hexactinellids.
See?
On such a tiny little stem, it's amazing. big carnivore sponges here, not the
Yeah.
asbestopluma, but different ones, right?
More anemones, so it looks like what we saw at the very beginning.
Yeah.
That beautiful rock with completely covered with anemones and carnivorous sponges.
They have such long tentacles, these ones.
Yes. Do you have an idea about the species of the anemone?
I can check if I can get an idea. Oh yeah, 1 minute till tape change. We'll change the tape soon, in less than 1 minute, but I'll let you know.
Yeah.
Oh, there are a lot of anemones that look similar here. Maybe Omatidae? They seem to have long tentacles. So when we change the tape, that means that we change the recording of the video, and we always save it after 2 hours to have backups in case anything crashes. We never lose too much data. And oh, can we stop the recording?
Yeah, ready to stop now?
This is nice.
Yes,
Yes.
stop. Yeah.
Okay.
And after 2 hours, we also always change the shift so that we can get a little break or continue, for example, lab work. And then Anne-Helene and Sergio will take over.
Okay, have to start recording.
Okay, start recording again.
now.
Yeah. Thank you.
This is really nice. Took a moment
You don't want to leave?
to change. Now we are here nice and steady.
Yeah.
Yeah.
Shall we?
Let's have a nice lunchtime.
Thank you.
I feel
Enjoy the
black,
dive.
actually. So
Just grab all my
can we go to the
stuff.
microscope?
Looking at sponges
Yes.
under the microscope. All right. Bye bye. Oh, lots of stuff flying around you.
Because we're slightly cold in here, right?
Yeah, but it's now 18 degrees.
Yeah.
Oh, wow. That's a lot warmer. Hi all, and this is Sergi and Anne-Helene fresh in from the lab. Directly from the lab where we have been looking at the samples from the previous dive, and we are still processing them. It takes a lot of time to get through all of the samples from a dive. Lots of noise in the background here, but we are continuing up the hill.
1,500 meters right now.
Yeah, we are about 1,544 meters. And as Hansi said, we have minus 0.5 degrees in the water. And that's colder than it was this morning, a little bit further north. Like 20 minutes of steaming further north, not very further, but there we had minus 0.2. A little bit higher up as well, that was, so not super comparable, but this is a beautiful wall, and it's almost a vertical wall.
Yeah.
Loads of anemone.
Yeah, we're still climbing.
And I am happy to click the anemone button.
Well,
And we see also carnivorous sponges here, right?
yeah, some of those carnivorous sponges, these white
Yeah.
branches, like the really thin branches that you see. Sometimes you see them, you see the lasers now, at the very base of the lasers, there's these carnivorous sponges. And there's a few there of them.
That are more branchy, right?
Yeah. We just have a closer look at this edge, where you can find these carnivorous sponges that you see. These
And
thin
also
branches.
it's Geodia, right?
Yeah, it's
No,
plenty
I
of
think he's creating
Geodia species. Oh, well,
once again,
some Geodia genshelli, which are these roundish sponges with the oscula,
One osculum at the top.
in the middle. Yeah, the oscula is it's a opening where the water flows after it's been filtered.
So the out opening.
Yeah,
What are the in openings called?
porus.
Porus.
Yeah.
That's the basic for the porifera.
Yeah.
Yeah.
There's some sponges you see here where the lasers are, which are pretty colonized by other organisms. If you see the lasers,
So that's a Geodia
yeah.
with carnivorous sponge
lasers have
and an anemone, right?
yeah,
And
other smaller sponges too.
and are all of those white tubes? Do we think that they are serpulids or?
Not the ones on the sponge,
But there
the
are some
ones
wormy things
beside,
on.
Yeah, definitely.
So that sponge is a whole little ecosystem in itself, even
Yeah.
before we start to look at the
Yeah,
microbiology that is also a complete ecosystem, right?
yeah.
So on very many levels, this sponge is an ecosystem. Okay, those 18 degrees were not that super warm actually.
Yeah. Okay, so
I really love all of these anemone.
they're bringing color to this
Yeah.
wall.
Together with the shrimp, there are these popping pink
There's a
areas.
sea star in here. You see orange one. We're still climbing. I'm still surprised on how these Geodia organisms they made it. I mean, they really like the vertical walls.
But these are some Rosellida as well,
Yeah,
right?
some Rosellida.
In between everything else.
I'm going too fast. So
I'm so happy I
fine.
found
I
the
think it's
carnivorous
fine.
sponge button because now I don't have to type all the time.
Yeah, there's a bunch of carnivorous sponges, meaning that there's lots of food for them,
Yep.
meaning crustaceans.
Ooh, you're rubbing it in again. And I just said from the previous samples that we took this morning, I've sorted out quite a lot of amphipods, tiny little ones, and I guess they would be the favorite food of all of these carnivorous sponges.
There were some big ones, right? Too.
Yeah, there were a few big ones, and most of them were in the family Pardaliscidae. For those of you who are really amphipod nerds, I think 1 or 2 different species there, which would be normal for this area. But already in the smaller size fraction I had 5 or 6 different species just in one sample, and we didn't even sample for them. This is just what comes with the sediments we sample, or with the bryozoa mats that we sampled earlier. And I think maybe we're seeing some of these bryozoa mats here together, maybe with spicules from
I
Geodia.
think those are the ones, yeah.
Because I can say, when I looked through those mats that we sampled, that they were a combination of spicules from, I would guess, Geodia, and lots of old bryozoa.
Yeah, we don't see the bryozoa, the living ones, right, in here?
No, so maybe that's just a spicule mat then, because there is no lacking of sponges here.
Mm-hmm. And just below, we see. We'll see in a minute. Sorry. This group of serpulids. These worms. They are building a structure in here. You will see in a minute.
Yeah, they're almost building their own little area, right?
So the white tubes that you see those are tubes of worms, a group of annelids, which are called serpulidae. And but there's some living, and there's some that are dead. The white ones are the ones with animal inside still, but they are alive. The other ones, the brownish organisms, remains of the tubes, those are the ones that are dead and are producing a substrate where all the ones have been. Oh, yeah.
What's the yellow thing on a stick?
Let's find out. I'm not sure about it. I'm not sure we have seen these before. Probably a sponge that got there. I don't think the sponge built this stalk. Probably colonized the stalk of an organism that died.
That's a good way of doing it. But this is also quite common in nature, that there is no unused habitat. If a habitat becomes available, it will be quickly utilized by somebody.
Mm-hmm. So you see at the right hand side there's some of the remainings of these tubes of serpulids.
Mm-hmm.
They're producing a substrate
Yeah.
for others to be colonized. And this sponge, you remember the name of this sponge on the top right?
Oh, that's an Oscornema foliatum.
Yeah. Yeah.
I am getting it. Now we have to just find the species of anemone, because there's so many of them.
Oh,
I think
yeah.
Fancy was
That's
trying to
the
find
one.
out. She had some suggestions and I didn't hear it, so maybe you guys already heard it.
Some shrimps. Yeah.
I think they're actinostolids.
Mm-hmm.
But there are many actinostolids, so who knows?
Mm-hmm.
Yeah.
Ooh, that's...
Can you wait a second?
Oh, this one we need to look at. I would
Joe,
think...
could put a move in please, bearing 045 at point 2 continuous, please.
Yeah, we saw something interesting, huh?
Yes, this is
Yeah.
bottom-living jelly. It is... Oh, I remember and I forgot, but I can find the name here.
So I'm gonna
let the ROV get stable. Oh, look at that! This orange
Yes.
stuff in here, we know now is not a sponge. We know now it is a solenogaster, which is a group of mollusks, which is not that common, I would say. It's more common in deep sea areas. Yeah, we're gonna have a look. See more detail. Really long. I was just measuring it after taking a picture, and it's like it was at least the one that we collected more than 4 centimeters, and I think this is one. Yeah, I bet this is.
So the let's collect some of those. And let's also collect the Lucernaria that we saw.
Mm-hmm. Look, it's moving.
Yeah.
Looks like a snail. It's not a snail. It's a mollusk.
It doesn't. It's not a worm, but it's a mollusk that looks like a worm.
Yeah.
Probably,
The Norwegian, yes, please, and the Lucernaria.
The one on top. Yeah,
Yeah.
we'll have a picture. We'll.
Let's first have a proper picture. Can we maybe also without the
Shall we slurp first this and then go
yeah?
with the
But
other
let's zoom in on it without the
Yes, it's just
with the
Lasers?
laser.
5.2.
It's
slow down a bit.
So on the right there's a worm, kind of a worm. Then we'll go for this one, which is really beautiful.
We first want to just film it a little bit, that thing that looks like a worm. And it is a mollusk, so it is close to snails and bivalves and octopuses and those animals, but this is covered by little scales, so it looks almost like it's covered by fur
Oh yes, please.
when you look at it in a microscope. Can we take off the lasers a little bit? I can do the notes.
Yeah. Yeah, that's the one.
The pencil is somewhere.
Here. The other one, you look at the
Yeah.
other one. I don't know. Probably there.
It's under here. How do we want to slurp them, right?
Yeah. I think
Do we
that's-
think that the Lucernaria will survive to be slurped?
I don't know. Probably. You can just give it a try.
It's a jelly
Ah.
thing, so maybe not. But this one definitely will survive to be slurped.
That's a very good fish.
Oh yeah, you cannot do it.
Go on with
Then we will just look at them. Okay,
Yeah.
can we go and look a little bit up then at the
We
Lucernaria?
have another one.
Yeah.
Please.
I mean, you can try, but it's just because it's vertical, you mean.
Yeah.
Just then have a look. Right.
There,
Ooh,
look at that. And if we manage to just have that in the picture a little while. So this is a jellyfish that is attached to the bottom. So this is a benthic jellyfish. It's called Lucernaria. Let's see if we can suggest.
So I'll stay here so that we can film it.
So the suggestion for a name in the area would be Lucernaria bathyphylla. And it's quite cool to see.
So it's like a jellyfish. What is that?
It's a jellyfish, but it is attached with a stem to the bottom.
Uh-huh. Let's have a closer look. it's got short tentacles, and in these tentacles they have these cells that when a prey gets closer and it touches it, it gets activated. The cells are activated and they sting.
So like all other jellyfish,
Yeah.
they have stinging cells, and it's also together with the anemone and the corals that we are seeing both on this dive and everywhere else in the world.
Mm-hmm.
What do you think that long, thin thing below it is? Do you think it's a worm?
Yeah, probably, yeah. Things below of these beautiful specimen.
Yeah, but that's a beautiful thing. It's like it has these little. This really has some small pom-poms, right? Going out, back in again. And you don't think it's possible to make a sample of this?
You could, I mean, it's vertical, so you have to free fly it and
Yeah. Okay.
move the arm as well. You can try that.
Let's
Is it
let's
slurp or-
Yeah, we'll be with the slurp.
Yeah, yeah, if it's slurp, I mean, you've got a fighting chance of you slurp it.
The thing is that, well, the slurp is going to be
I think it's
The
going to
thing
be
is
quite destroyed if we slurp
damage.
it.
Yeah.
If you bring it out and free fly it in, you should be able to get it.
It's fine because they're so soft. And then when you slurp it, then probably it's going to break into 1,000 pieces.
Would it be a scoop or?
A scoop would be the best, I think.
Yeah.
Hmm?
Still looking at the comp there, just to see if it's.
So this is really an interesting little place right below the, and so on a little ledge between two vertical walls here,
Look
right?
at this beautiful wall.
Yeah, look at that
Just
wall right
waiting
above.
for us.
Yay.
Is it on there?
So we're going to try to...
Now, for all of you who were rooting for the University of Bergen Manet team this morning, when you were saying we should try to sample ctenophores and stuff, this would be this is a picture I already sent home to them. So we are thinking about them. Whenever we see things, we think about all the different colleagues we have that would like to work with these animals we are seeing. So this is definitely a collaborative work. Whenever we find something and sample something, we don't only sample for the 6 of us, 8 of us on the vessel, but we sample also for colleagues who are working on specific groups. And that even goes into the nerdy little details of that genus is a specialist genus for that person. And sometimes it's big groups, sometimes it's small groups, but this is how the entire community, I would say, of taxonomists work. There is so much life to work with and catalog and discover that we don't need to fight about it. We can we all have our specialities. And we still can collaborate very nicely, so.
Yeah.
Stay fresh, jumping arrows and be there. Wait till you scoop it.
Yeah.
You wanna hit the rock and you sit there, flick it from underneath.
Yeah.
Then the rock just pushes off.
So yeah, we are considering which is the best approach to get the sample. This is not. It's quite challenging in this case because it's almost a vertical wall. Then the ROV is standing, it's flying. We're gonna try to get it using a scope because probably with the slurp it would have been easier to collect, but we would definitely damage the specimen. This is something that we don't want to happen.
Yeah, but that's an amazing animal.
It is. Just now that we are
So still, it's
Yeah.
very nice to zoom in on this one, yes.
By mistake. Yeah, he's used to this as well, so I'm not.
If you drop it, it's a long way.
Do you want to do it?
Yeah, go sit here.
just right in the center. It's a beautiful
It's sitting
specimen.
there pretty much stable up
Yeah.
12% first one, but we can try, just try and leave it just doing it. I can go ahead and scrape it. Yeah, it is.
So again, our amazing pilots are going to give it a try.
And you might need to start where we are.
You want me to zoom in the...
Yeah, I'll just get it scooped first.
Yeah.
Okay.
Is that attached to that rock, do you think? Or
Probably... yeah.
Maybe, yeah.
I'll have a quick look first, rock.
Okay, and then move on to stop.
Well, wait, I'll have a look at this rock first just to see if it's attached.
Ah, yeah.
Rock.
It's probably not attached. Well, it's probably attached to the rock.
Can you zoom in on your camera shot, please?
I think we're way too much in the
And go to
front.
the
Oh,
right.
yeah.
Oops.
There's like a really small stone there.
Oop.
He didn't like that,
Uh-huh.
but that's okay. We can bring him back.
Oh,
You are sampling like you're picking flowers. Oh, no.
No air. It's attached to it. Just go on it. Try to get the smallest of grabs as well.
Oh, it's flying now.
Ooh.
It is. It's fine.
Ooh.
I catch.
Oh no. Oh, oops. Sorry about that.
Flight. It's in the front there.
So it managed to escape.
Yeah.
Well.
Yeah. Okay, these creatures. They,
Well,
they know
it
how
deserved to stay.
Oh look, it's flying. It was flying on the left hand side. Yeah.
So okay,
Don't worry.
Didn't know where to come with.
we got a beautiful film of it, and it will find another rock to attach to.
Mm-hmm. Okay. No problem.
Thank you for trying.
Now we are at the base of this
To the laser one?
beautiful.
Yes, please.
Lasers on.
And let's fly up this very beautiful vertical wall.
I know,
Got
yeah.
it the first time.
Trust us.
Okay, all the species buildings, Lysodendrorix, the anemone, sea stars. This is a community we saw yesterday, right? In the Schultz Bank. I think so. So they were, look at that. Look at this density of anemone.
Yeah, look at that.
Let's just stay here. For a sec. That's indeed really beautiful. There's some Rosality also here.
I am going to dare push my anemone button.
Mm-hmm. Yeah.
I did it. I'm going to do it again.
Deserved.
Another. Just to indicate we have quite a lot of them, and some carnivorous sponges.
Mm-hmm. Yeah.
And my eyes see Lysodendrorix.
Mm-hmm. Yeah.
Yes.
It's been a
Do I
minute
win the
as well.
apple for this round?
Yeah, but what's the name of this beautiful creature? Luternaria bathyphylla. Yeah. Bathyphylla meaning that it's a really deep organism.
Yeah, it likes
right?
to be in the deep. Bathyphylla. Loves the deep.
Mm-hmm.
Oh, but this looks almost like a painting by
Yeah.
Van Gogh or something like that, right? This is really...
Yeah, super beautiful.
You know, like that Starry Night from Van Gogh?
Mm-hmm.
Van Gogh?
Yeah.
Yeah, the Starry Night. It's a
Yeah.
little bit... If
Yeah.
you made all the stars pink, I think it would be there.
Not smash it completely.
Is that the tulip sponge or is it something else?
Yeah, I think it is.
Tulip sponge?
Yeah. The
I'm
very
definitely getting the
base.
apple of this. Oh, and a fish.
It's found.
Right, John, if you can all start now, please.
It's found a place where to hide.
Yeah. But this is a good place to have a lot of cracks and crevasses and everything, so you can hide in there. And of course, we have tons of geodia. So are all geodia kind of smelly when they come on deck, or is it just the shallower? So
I
like
don't know.
geodia macandrevi.
So Macandrevi is producing smell.
Oh yeah. Or you can smell it on the entire boat when you get a load of those up.
Yeah, it means that they have like chemicals, right? And Paco, the other sponge person, so we have Julio, Paco, and myself studying sponges. So Paco is interested in studying the chemicals that sponges produce, which may have in some cases, some application for new drugs or new medicals.
Yeah. And that's probably why he started with Geodia then, because they definitely, if you have a lot of Geodia on deck and it's a lot of weather, you can become quite seasick
Yeah.
just from the smell.
You see some of these anemone are with the tentacles inwards. So those are the anemone.
The orange
These
balls.
in here, those are anemone with tentacles. Inside the
cavity.
Yeah, it's the gastro.
Yeah, because
gastric
it's one
cavity.
cavity. They have just one.
Yeah.
It's one opening into the cavity, and that's where the food is processed.
Mhm. So they have a proper anus or
Well, it's the same as the mouth.
yeah.
In and out the same
Simplified.
place. Yeah. What's that long thing?
Yeah.
Is that another carnivorous sponge?
Oh, I don't know. Probably this is something on top of sponge or something growing on top of this stalk. Could be carnivorous sponge. There's plenty of those around.
Okay, I will hit the button for carnivorous sponges as well, and for Rosellida. Another Lizardendorix.
Oh, yeah.
I'm just waiting for you to tell me where my amphipods are, since I'm telling you where your sponge is.
Everywhere.
Everywhere. It's easy. So all of this, that looks like sedimentation. Do you think
brownish?
that? Yes.
I think it's the result of these pulids dying, and the tubes that they produce are like getting in there.
Mm-hmm. So all of the white dots you think are
Yeah.
serpulid?
Yeah, indeed.
Yeah.
To make it closer look.
The small white dots we're talking about now.
Yeah. There's plenty of those. And they're producing like a kind of a micro-habitat where I'm pretty sure there will be many different small creatures living.
Yeah, I'm very sure about that too.
Yeah. There's some Asconema foliatum. There's another anemone there with the tentacles inwards. Most of them are like with the tentacles everted, trying to find food. When they get food, they get the food with the tentacles inside the cavity where the prey is going to be digested. Yeah, we're like going upwards to see whether we reach the very top of this mountain.
So the plan is to get to the top and then follow the ridge a little bit again like we did last time. But this wall is so pretty that right now I'm hoping that it will never end. It's also super interesting to see this community of animals together. So very rich. See there? Amphipod going down on its way to food.
So need to forget that we have to take our eDNA samples when we reach the very top.
Because one of the things Sergi and I are doing here is, in addition to finding out everything about the sponges and specifically about lizard and dorix for Sergi, and trying to find out about the smaller animals for me, we are also trying to find out the environmental DNA, so the DNA that is left in water or sediment or filtered by natural filters, such as some of the sponges from Sergi. We try to find out this so that this is a less invasive method, so it's still in a let's see if it really works stage, I would say. And one of the reasons it's still in a let's see if this really works stage is that we need to build a library to compare the environmental DNA with, so that we know which species we have in the environmental DNA, or eDNA as we like to call it. So we take samples of water and sediment at the same place so that we can compare as well. And then we find out which DNA is in this sediment and water, and we compare it to what we already have of libraries for species from this area, so DNA libraries for species from this area, and at the same time we're trying to build out that library further, because we don't have anything specific for the mid-ocean ridge or the Arctic mid-ocean ridge yet. We don't have anything even specific for this whole sea, this larger area of the Nordic seas. Many of the species that were found here historically have maybe not been refound, so we know of species names and some.
That's a very top.
That's
right?
the top, yeah. Some descriptions, but we don't know their DNA. So that's one of the reasons why we're collecting all of these animals. It might seem a bit obsessive to sample all of these things, but it is to make sure we have a good library of both which species we really find here, and sometimes we do need to look at them in the lab to decide properly which species this is, and also we try to then use these animals to, sometimes we need to make a redescription, sometimes we just need to identify it to the correct species and say, okay, now we get the DNA from this species, and sometimes, quite often maybe, it is an undescribed species, and then we need to describe it first. And modern descriptions, of course, normally now always include a DNA voucher as well as what we say, morphological or a physical voucher at the museum. This is a beautiful rock.
Look, this is a pinaronic.
Ooh, yeah. Sea spiders, here we come.
Oh yeah. Okay.
So now we are at the ridge, and it's a completely different fauna. We have
these small anemones. It's
small
soft corals,
anemones and soft coral, mainly soft corals, and then some Rosellida.
Those are not the same ones as the ones we've been.
No, these are some other ones. So we should maybe also try to get a sample here.
Those are really small if you compare with the pinkish that we've been seeing in other areas.
And all of these long thin things that stick out, are they all carnivorous sponges?
Yeah, indeed. So we need to. I think we should take a small sample, right? How about taking a rock, small rock? So we're gonna try to take a rock with this soft corals.
If we can find a small enough rock that it's possible to bring in.
Oh yeah. I don't know how big is this one? What do you think? Is it too big?
Which one? Can you
12.
point to my screen here?
What do you think? Is it too big?
Yeah, no, okay.
Is that the one? Yeah, that's the one.
Yeah, it's a little bit big, I think. It's
or even
like-
a smaller one. Ah, yeah,
yeah.
here. It's somewhere here, one of those mix one, or even there. Yeah. That's probably too big, yeah. But let's just first
Let's first
Enjoy
just
a little bit this.
enjoy a little bit and zoom in and look at some of these.
Can you turn off the list, please? Again, and this is the
Yeah.
solenogaster.
The solenogasters are everywhere. I have
Uh-huh.
never seen this size of solenogasters before.
This is the orange, wormy like specimen that you see just below the lasers right now. Well,
Which one do you want? Because we got quite an angle here. So
I think this one. With the scope. Can you turn off the laser so that we can have like a still image, like a
Thank you.
I'm not going to turn it.
of this habitat? Interesting.
So I need to change position
Yeah.
somehow.
Yeah.
Ah, yeah. Tilted.
Yeah.
That's another one. These solenogasters. Probably another one in here. It's plenty.
And there.
I mean, just seeing solenogasters everywhere, right?
It's like when you're learning to
Much.
observe a new thing, then all of a sudden you see it everywhere. Is that the sea spider again?
Sea spider, yeah. There's quite a few of these.
There.
spider. You see?
Yeah.
Uh-huh.
Okay, so we are in the solenogasters heaven.
You get it from that?
So, okay.
Trying to position the
Not-
ROV. I think it's going to be a really challenging taking samples from the sediment here because it's mostly rocky.
Yeah.
Do you want to come further back?
Just where is it on your screen again?
This one. This is one. That's the one you just pointed to. Yeah.
That in just in front of you.
Was this one you said?
That one here, this should be like
Right, close on the screen here?
yeah,
Yeah, that one, yeah.
for sure.
Do you want to come further back?
Well, Metern Tiefe as well. Is it-
Yeah, the west one you
But I got-
scoop.
Scoop.
I think
rock
the
on
scoop
my
would be
left.
enough, right?
Oh, there's like a rock in my left.
I think the scoop would be the best, yes.
Yeah.
One we've got there, and a rock there. I can come further back.
Metern.
Yeah. Move back towards more left.
Aye, we're still 6 degrees here. A good bit off the front. I that came away.
Oh, yeah, see for your height.
mind going down and looking around.
I can come back.
I know it's near.
Oh, actually.
The moment in front of you there.
Oh, yeah. Oh, there you go.
Could you do it from here?
Yeah.
What could you do it from here?
Yeah, just to see if you can reach it.
So
In front
one
you,
of the difficult things with sampling is, of course, that the two manipulators that we have available need space, and we need to sit at a good place. And the things we want to pick up are normally a little bit bigger than
Yeah.
we think they are. And look at that rock on the side there with all of the carnivorous sponges. That's crazy.
So you know how to use scope?
in front of you.
And we are sitting still on the edge of a hill.
Scoop for this one, yeah?
Yeah.
Yes,
I think
please.
so.
So I'm hoping we're not tipping over. Gives me a little bit of dizziness to look down that hill.
So which ones are free? Top left.
I think everything apart from
Top left.
I was going for the white bells.
Do you want it in? Yeah, okay. That was a beautiful shrimp swimming by in a quite peculiar swimming method. So, if we are on the top now, are we also... No, it's the same transect, right?
Yep.
Yeah, okay. Okay, so while we're now at 1,445 meters depth, the water temperature is minus 0.4. So slightly warmer. Not really still swimming temperatures, no. But it's also slightly different salinity to previous, so it's obvious that we are in a little bit different water mass, which is what we have been expecting since we are on the east side of the ridge and not anymore on the west side of the ridge.
Which one is potentially also be this one if you want. So I cannot reach to this one with the camera now.
Okay.
If you want to go for it, then we'll be able to see the.
I mean, if you want that one, we can like.
No, it's fine.
move back
So this one is fine. I mean, the one that you think it's the easiest.
Yeah.
This one is closer, I think they are easiest.
The one
Yeah.
close.
These
Yeah.
ones.
Yeah.
Already here, just.
So. Yeah, and it fits in the scoop.
So what I am also seeing from our log is that we have
Does it?
previous sample was exactly 1 hour ago, and we have
Very nice.
moved 100 meters up the hill in that 1 hour, if you are wondering which speed we are moving at. So 100 meters in 60 minutes on a more or less vertical wall. I think Usain Bolt would have done it a little bit faster.
That was one of these soft coral, right?
Yeah.
Lion.
So yeah, this ground is very hard here.
Yeah.
I don't think we will try push core.
Not here.
This is not going to be the place.
Is it attached to the other rock?
I think it's just a part of the bedrock, more or less.
If it's not loose, just leave it.
Yeah, don't try to take the whole mountain.
Yeah, we thought it was loose. Yeah.
It's too big. Probably it's
Yeah, it can be right.
really big.
Don't worry.
The balance of plants in the weight. Pick up that rock.
Okay. Then,
That's a drop in the
yeah, don't fall down now.
Should we already
Let's go on.
It's too big, this one.
Yeah. Wow. Indeed. Yeah. Don't worry about that. We'll find another spot.
Starboard go ahead.
Yep.
So we're gonna use the scoop now, but pretty sure they will in the future.
Yeah. So these big white sponges in the background there, they are again the Rosellida?
Yeah, I think so.
Yeah?
This is as far as we can go for now. And
But
probably thanks to
this,
Leo and
yeah.
Paco.
But at this base, they have not decorated themselves with the crinoids.
There's no crayons.
So they are not as fashion conscientious. Yeah?
Yeah, we haven't seen any, right?
No,
do
the
you
crinoids
recall?
are sort of missing here. But we do see some of the Acidia every here and there. We see, and remember those Acidia, they are our closest relatives in this area.
Look at this.
Yeah, look at that
Huge.
sea spider. He looks kind of tentative about the whole thing. Slowly going around.
Yeah, I wouldn't say it's in a hurry, right?
No, I read one place that they are pushing the blood or the hemolymph around in their body using what is sort of more or less the equivalent to farts. So they are
Okay.
using gases from. So they're farting the blood around the body. Which might not be too polite to talk about, but it's Monday, so it's okay.
Absolutely.
The beginning of the week.
So we have these kind of quite large. We can run it.
Lasers on her.
Yeah. Oh, it's,
Oh yeah, look at that. So between those dots, my friends, it's 10 centimeters. And between, so that's 15, right? From tip of leg to tip of the other leg.
And hold on 1 second, because we want to talk about these again.
You want to talk
These
about the
mollusks.
Solanogastrus,
Oh, I cannot go.
the
further.
worms, the mollusk, the
Yes,
wormy
yes.
mollusks that you see at the base of that soft coral there. You see an orange little worm thing
Wormy,
sticking
yeah.
around, and that's a mollusk.
Solanogaster. We collected 2 in the previous time.
Yes, so we already have some of them in the lab, and
Can continue with life.
yeah, we will bring them. So What is that? Is that a jelly? The red thing?
I think it's shrimp.
It's a shrimp
Backwards,
going
right?
very slowly backwards. Yes,
Uh-huh.
it is. It's just behaving like, oh, there it's behaving like a shrimp again.
So we couldn't sample these small soft corals.
Because they live on
Here,
two
but-
large rocks. And they're everywhere.
Uh-huh. Plenty.
They're standing like little choir boys all along there in a nice little row.
Yeah. Mostly on the edge of each of the
Yeah.
rocks. Oh look, fish.
Ooh, a nice fish. So this morning we came over the sleeping ground of these fishes. They were all curled up sleeping. And we were thinking maybe that would be nice to do for us as well, just curl up and sleep. But there has not been a lot of rest since then, because we did sample a lot on the previous dive.
Yeah, we are on a shift.
And the lab is full of samples that need to be sorted and preserved and registered, so that when we bring them home we know every detail we can know about them and we don't mess them up with anything else.
Yeah.
So when you see us making all of these, or hear us making all of these notes everywhere, it's because we need to know exactly where a sample came from, and so that we can correlate it to the video and to the information we have from the CTD that is on the RV. And yeah, all of this data will go into the databases that we have in the different museums of that, where all of this material will go to in the end.
Yeah.
And we will
keep this
spread it
data, yeah.
between several different museums. Quite a lot of, actually all of the sponges, most of the sponges will go to the Museum of Evolution in Uppsala. Some will
Part
go,
of them will also
go
to the
to-
National Museum of Natural Sciences in Madrid.
yeah, that's where you are working.
Yeah.
Yeah. And then the small other animals will be shared between the Senckenberg Nature Museum in Germany and the University Museum of Bergen in Norway. So when things are in museums, they are available for science because they are then listed into these collections, where it's possible to either come and visit and study the collections,
Yeah.
or sometimes, maybe not so much lately, it's becoming difficult to send things, but it used to be possible to ask for loans as well. But shipping specimens is quite a complicated thing.
Oh, there's a fish there.
Is it a fish or is it just a shape? I have been wondering. I think it might be just a shape. No, it's a fish, you're
It
correct.
is,
Ooh, your eyes. I left my glasses in the lab.
Bit
This is my
far, but
excuse.
yeah.
My glasses are not here. I don't have anything in front of my eyes. I don't see a thing. I kind of
Okay,
got a little bit
yeah.
there.
Underneath the fish. It's just
So-
center. Oh, sorry about that.
You were going to check how high this wall was, and it's the ridge, so it's a hill on the ridge.
There is no soft sediment we can use to put
Not right
scores,
here.
Right?
Let's see what's behind the hill on the ridge. But is that round thing a geodia?
The one here.
This
yeah,
one there,
yeah, the one
in the
with
middle of the picture?
Yeah, the one with the lasers.
And
think so.
yeah, the lasers and the hole in the middle, the
Yeah.
osculum.
I think so. There's some fun shapes. Just we collected a piece of it, of one similar organism during the first dive, and we need to identify. We will. I mean, Julio Cargo will shortly identify it to species level.
Yeah, Paco is our Geodia hero.
Ridge RV.
And oh, look, there is again all of these going along the ridge.
Yeah.
Okay, would like to have a new move
everything.
direction north at 0.1.
So here is a combination of soft corals and carnivorous sponges and an enormous sponge of
Okay,
something.
thanks.
And that's a hexactinellid, right?
Not
Ned?
this one. I think
No?
this is not.
Ooh,
the other ones
look at the
are
shape of that ridge we're going along.
probably not this one. Yeah, that's beautiful. I mean, these small soft corals.
But also look at the landscape. It's
Yeah.
crazy.
It's quite different from the previous dives in other areas.
Yeah.
Like,
I am wondering how much pain it would be to go for a walk in this if it was above sea.
yeah.
It would have been quite steep. And also, just a reminder, this is a seamount next to, on the side of this mountain ridge. But now we are on the east side. And this almost sheer wall going down to the west. So we're going towards the north now along the ridge of the mountain. And there is this wall to the west,
Yeah. Can you go a little bit faster?
which is quite
Sorry.
interesting.
Faster, yeah.
Yeah.
There is another fish hanging out there in amongst all of the soft corals.
Yeah.
And they just make the whole mountain look fussy.
It's not that they cover the entire rock. They just cover the part of the edges. That's
Yeah.
where they get more higher access to the water column.
But it's also interesting to see they're all facing east, so
Yeah.
the water must come from the east or from the south or something where we
Yeah.
are coming now. Because they are trying to get food from all the particles that are floating around in the water. And seamounts such like this are making a lot of little mini currents around them, because the general current is coming from the south to the north in this area. And whenever there is a seamount or anything, it will make a little extra diversion for that current, so that they will have little eddies and little
Bridge
curls and
RV,
whirls and
can
stuff
we
like that.
speed
So
up to 0.2?
we will always have lots of extra little twirls and
Yeah.
Okay, thanks.
difficult to imagine beforehand currents on these seamounts. And this ridge is a little bit up and down, right? So we will do a little bit of a straight to the next hill.
So it's been 1 hour since we got here, right?
Yes,
Good.
1 hour, and we have been going up. Ooh, yeah, we got to the top, and now we're sort of more flattish.
So we like trying to get on track again with this transect that was it was Jenny that was thinking about this transect, right?
Yeah.
She was the one planning the way.
So we have done
to
the
go.
wall climb, and now we will follow the ridge a little bit. And we see the ridge stretching out in front of us with little sponges or big sponges acting almost like beacons in the deep here.
bridge RV, can we increase again to 0.3? Okay, thanks.
Yeah, definitely different environment, despite being at similar depths that we have already explored in other areas. So yes, you never know when you are diving in a place.
Yeah, this
What are you
is
gonna find,
completely unexplored, and it's not even having a name.
Mm.
So seamounts are getting names when the official maps from, I think JEPCO is at least putting out one map with names. But then the different countries are also putting names to their seamounts. So this one, so far, is unnamed and unexplored until today. So now we're trying to do a little bit of exploration to see what it looks like.
Some short neutral points in the world.
Quite a difficult one, this one, because it's got a little bit of ups and downs.
Yeah, and it's also because the background map that we have is not, the resolution is not super good, since this is quite unexplored. So, I mean, well, it is super good for being in the deep sea and everything, but it's not, I think it's 50 or
This
30.
is more muddy, isn't it?
Yeah, maybe we can get close and see.
Yeah, because at some point we would like to get these push cores on Niskin bottles on our sponge around. What are these sponges? Okay, these are fun-shaped sponges we could potentially grab. What do you think, Annalene?
If it is muddy, that would be a good place.
Do you think we could land here? Where now? Somewhere around here, so that to
And check if it's possible to do a push core. Let's first check if the bottom is
Yeah.
available for push coring. But it looks like it at this distance.
Yeah, pretty much.
It's a little bit rocky.
Yeah, to get closer, you see.
Yeah, that's the thing about getting closer. You always see more details when you're close. But we have the Seriantids in there, so it must be a little bit deep, right? So we have these tube anemone. That is a good indicator that there is a little bit of sediment. Yeah, let's try here. And make a feel for the possibility of a push core in between the cobbles.
Yeah.
Oh, look, there is. Do you think there is a burrow?
Yeah.
That's beautiful.
It might be the right spot.
So let's zoom in on a couple of things here.
Like this oriented, right?
Yeah. Let's look at that one. And look at all those little orange dots.
Those are the
Anemone?
anemones.
Little orange anemone dots.
Yeah.
But then around them are these are sponges, right?
Granulas.
That's craniella.
I think these are the granulas.
The brain sponges. And then anemone.
Yeah.
Well, this anemone lives down in the sediment and can subtract down in its tube when it does not like to be up on the surface anymore.
So we're gonna try to get samples from the push cores, like somewhere, I think.
Yeah, somewhere there, right in front of the. Yeah. Can you just quickly zoom in on that thing that
Reach
might be a
LV.
barrel?
Can we change the direction to 050? Okay, thanks.
Yeah. There's a barrel here.
But we don't see anybody at home. And it's covered by sponges.
Yeah.
That's a fancy house. And an opening of almost exactly 10 centimeters, huh? That's a big opening.
So let's just, sorry, I'll go down.
Now we are going to look at the push cores.
You also have the max, remember?
Yes, we do
They
have
were
a
not
max, yeah.
used so far.
What was that push car number 5?
Happy anywhere?
Happy
Yeah.
anywhere in
Yeah.
this area.
Let's try. Not entirely sure whether this is soft, really soft. It's soft, indeed. Thank you so much. Yeah. Yeah, got it.
Okay. And that's number 5, isn't it?
I think so.
So these samples will be for environmental DNA or eDNA.
Yeah.
And it's quite
Great.
muddy when we get under the rocks.
It's number 5, right?
Yes.
Just this one. Yeah, I think it's fine.
Two push cores here.
Yeah.
2 push cores, please, yes.
Yeah, 2 replicates in here.
I think we have both coffee cups left.
Same kind of area.
Yeah,
Yeah.
not super close.
Here, okay?
Yeah.
Yeah, that sounds perfect.
Ah, the anemone was like getting disturbed.
Yeah, it
Yep.
dove down into its hole.
Bridge, LD, can we stop for now? Bridge, ROV.
Yay!
So to push chorus.
Can we stop for now?
Stop.
Okay, thanks.
Now we need to do the Niskins.
Shall we do also the marks?
Let's do one more here as well.
Or we're gonna have marks here too. The marks.
Yeah, but let's do the Niskins.
Yeah. Can you trigger them? The Niskins?
Niskin, yeah. The Niskin.
Can I? So the bottom one. Yeah, worked.
Bottom.
Top one. Looks right.
At least the bottom is
The
paying right.
bottom work.
Yeah.
This one.
Top one, the top plugs definitely, and the bottom one, should be just take a look, keep pushing.
So we will use the bottom one on this.
Yeah.
So the ROV has two Niskin bottles, which are like bottles that can be closed at the depth that you want.
So
And
it's
then-
like a tube that is open at both ends until we close it. So we have the water from the...
This exact point.
Yes.
Yes.
And then we just close the top and the bottom of the tube, and then we have how many liters?
I think it's like 6 liters.
Yeah, so then we
something
have
like
a
that.
6 liter sample of the water right where we are when we trigger the Niskin. So that's a way to get the water samples, and both water samples and the push cores are going to be used for the EDNA. And now comes our cutest sampler, that is the coffee mug. It would be quite difficult to drink coffee out of this because, as you can see, there is a net at the bottom of the cup instead of a bottle at the cup bottom. So we use it to sample sediment into the
Close to the
bag.
area where you also did
What's the
not the same ones,
but
close.
close to,
Close to where you did the push course, but not
not on top of it.
Oh, yeah, I'm sorry. I think you are right on top of one of the push core. Yeah, that one.
Yeah.
A little bit further if you want. Yeah. Like here. Yeah, that will be good. Oh, there's a rock or something. We can go somewhere else.
Set as noon and we
to go back to the door. Try and do that.
the idea is that we're gonna have small representation of the sediment in this area. It's gonna be filtered, and hopefully we'll have a bunch of
Amphipods.
amphipods and other creatures that live in the.
Yeah, we want other creatures in addition to amphipods. I'm not going to be against other animals. We want them all.
Yeah.
But so far in these samples we have found lots of little worms, a few crustaceans,
You can take a little bit more time if you want to clean it up.
and some mollusks. These are the main groups we have found in these samples.
Yeah.
Of course, we're seeing on the soft sediment, we see the seriantids, these anemone that are pulling down into their tubes. And we see sometimes, but not here, we have seen stalked both crinoids and stalked sponges, but right here we don't see them.
Nope.
So this is a different
That's quite different. I would say.
fauna, yeah.
Some different species.
Oh yeah, look at that sediment sample. That's going to be a joy in the lab.
Latte, that looks like, doesn't it?
Are we all dreaming in about an espresso right now? Or is this a tea gang? You're the coffee gang.
I am, yes.
Yeah, we have the coffee gang. Oh, and we have one tea man back
This is
there.
latte. Or an Irish coffee, I like
Yeah, it's quite thick. It's nearly going into the
Oh
bottom.
yeah, it's fine.
Yeah,
You can just put it back into.
it's definitely an
Firebox.
espresso, a double espresso.
So it was top right. Right.
Yes.
So it's gonna be a little bit
Oh,
of
it's
work
a-
in here.
Yeah,
to be
that's a
done.
big sample. It's gonna be, but 11, 12. We only have 2 hours left of our shift, so this is work for the other group.
To keep them busy, right?
Yay! To keep the pressure up.
Yeah, we need to have some more samples. Yeah,
Yeah.
they will be happy to be working on this material. Oops, there's
So one thing you can easily see here is how strong those manipulators are, those arms. They are very strong. Do we think we still can sneak down the coffee mug? Yes.
yeah.
They're also very good at doing very detailed work.
Do you think we should take another one?
Not here.
Not here. Okay. Thank you, guys.
Thank you so much.
Then, ideally, can you?
Did you want to do a second coffee mug?
I think we will do a second coffee mug another place so
Another
that they
place,
will
okay.
look a little bit different maybe.
Okay.
So
At
ideally
least that
now,
will be the idea.
turn a little bit to the left because there were some sponges. You want
Right,
to grab
okay.
a piece of sponge?
You're going to grab some sponge,
Yeah.
okay? Okay. Just draw. See if you want a job. Right, sorry. And you're going to grab a sponge. Yeah.
And this sponge we will use as natural filter also for the eDNA. So
Yep.
that's why we want the sponge from close by here so that we have all our eDNA samples from the same place. That makes for the
Do you want to spend
best
it to grab it?
setup.
No, that's fine.
Yeah, you can.
You can just.
Where did we have the sponges?
I think they were like on the left. These sharp, fan-shaped sponges.
Oh yeah, you want the fan-shaped sponges for this. So Sergio has been working quite a lot with finding eDNA from sponges, from natural filters.
Deep water and shallow water, and sponges they perform really good at collecting the DNA.
Yeah, well,
of the
they're
surrounding
here all the
habitat.
time filtering, so something is, now you have to help them find your sponges.
Where do you want me to go for this one?
Yeah, just go up a can tilt for the left. No, not this one. There's a few in here. So
I think we have to lift up and turn
Yeah.
around a little bit. Because it's that the audio?
yeah, but the geodias are not the best at filtering. Sorry. So there are two main groups of sponges: the low microbial abundance species and the high microbial abundance species. High microbial abundance species as Geodia, they can be like more than 90% of the tissue of the sponge filled by bacteria or archaea, which is like a crazy amount of microbes in there.
So they are an ecosystem in themselves.
Whereas the low microbial abundance species, these fan-shaped sponges or Lisandorix, Complicata, and many other ones, they have like a less amount of bacteria and archaea, and they are like more active filter feeders. So this makes a difference because the geodia eventually is not filtering as much as other.
Oh, because it's just hanging out with its bacteria instead.
They rely more on the bacteria. This is a symbiosis with the sponge, and they get food from the bacteria. Whereas the other ones, they have to filter more actively.
So is there an evolutionary, did they split up and then one side became
High
more-
and low. Yeah, that's a good question. I don't think so. But there's like
So it's
some
something that
sponges
has
are-
happened several times
Yeah,
in
sorry,
both.
I'm looking for sponges.
Oh,
Uh,
so Sergi,
I think
Sergi, you
there's
have to help them look for sponges.
So if you fly a little bit up.
Yeah, no, I can fly up. No,
He has been moving around. Do you see any? If you
Not
zoom
really.
out.
I think they were like closer to the edge on the left hand side.
We are on the edge,
Yeah.
so I don't think we can do a lot more.
I think they were a little bit backwards. We can turn it.
Turn my head to left.
Yeah, to the left. I'm probably a little bit backwards.
There's a list of Dendrix there, isn't there? Because then we would be able to use that one, right?
Yeah, if it's less than Nordics, yeah, that'll be.
And now we have a dust cloud.
There's something
Yep.
there, but what's that?
Is there anything?
Not sure about it. I think this is one of these Asconema, right?
I think so. Are you asking me about sponges now? Yes! I'm at the level where I'm being asked about
This
sponges.
is not the most interesting one.
Okay.
If you turn a little bit and go backwards, because this is an exactinelid, and they don't
They are glass sponges.
Yeah.
They're made out of glass.
filter that much as
They're just standing there being pretty and made out of glass,
yeah,
huh?
that's it.
Ooh, don't touch me. I'm made out of glass. I don't need to filter. No, but they're super pretty.
Yeah, they are.
And very interesting that animals can be made entirely out of glass.
Almost 10 rotation now. 0.8.
in my head except for
Well, now we're really into the cloud, huh?
Yeah.
So this is the thing about being in a soft sediment when you are looking around. We do stir up a little bit of the sediment.
I've got to watch my turns to set the time. To the time.
We don't want to get knots on our umbilical, right?
No, we don't.
That would be very sad. So of course the umbilical or the
to clear this.
cable that goes from the ROV and all the way up to the ship
I see something. Yeah.
is very, very important to take good care of. That's our
That
lifeline
sponge
to
that I was
from the ROV to the. That's where all of the communication with the ROV is going.
I think this is the one.
Both pictures that we're seeing and all of the information that the pilots are giving the ROV about where it should go and how it should
Should do the
behave.
trick.
So it's an amazing piece of electronic hardware.
Is that a sponge or a rock?
I'm not sure anymore.
It's a rock.
It's a rock.
Rocky sponge.
Okay, let's go and see if we can find something that is
Yeah.
close.
Otherwise,
Because-
yeah, it's.
Yeah, we're just kicking off.
Yeah, I think we just have to try to see if we can get somewhere
Yeah,
out
we'll
of the
have
cloud.
another one close. I mean, it's
Yeah.
the same habitat and.
Let's just moving forward. Yeah.
Sorry about that.
No, that's okay. And then we get up to this.
Yeah, let's just get out from this.
Out of the sandstorm and hopefully without any bows and ties on the. There is another spider.
Yeah.
Yeah. And look, it's another hill right in front of us,
Uh huh.
like
If it was
a wall. And
These are
Sergei
Escanema.
immediately goes for looking at the sponges on the wall.
Asconema. Yeah, I think it's Asconema foliatum.
Anything, yeah?
Yeah.
It's the wrong type of sponges.
Wrong side.
I mean, we could.
So now
potentially
you're
grab it.
picky as well.
Yeah.
Yeah. All sponges are equal, but some are more equal.
Yeah, we're not finding the right one. Yeah, these sediment all over the place should be cleared. Yeah, currents are gonna do the job.
So this is definitely wall again,
The
right?
wall, yeah. Just beside the area where we're collecting the samples, the eDNA samples. Yeah.
But we made the whole area quite sandy.
Mm-hmm.
I think that one is the one that we were coming from. That wall was the one that we were coming from.
Just trying to find the right sponge in here.
Down. We were going down.
Oh, you think we are going back to the same place?
This wall, I think so.
Uh-huh.
Because we were going down and then we just move here and then we are.
Oh, then we need to actually sort of go the other way.
Yeah. That's status.
Yeah. Gotta be patient.
eliminated basically because we are moving to the same direction. I think.
Oh yeah, those are
Side turns. Oh yeah, no.
yeah, those could be
Yeah,
a potential target.
Me to turn my head all the way around.
Oh,
I think so because we were coming from here, then we this loop and now we are coming this way. So
this face. It's coming.
Yeah, all of a sudden it's all of the sponges you don't want, right?
Yeah, exactly. Nelly. It's
should be roughly 0.7, wasn't it?
so if you spot one of these fun-shaped ones, let's give us a shot. Is it the one?
The thing is that
Well, that's a big
we need
one.
to go the other way because we are coming again.
But I don't think saying that we are going the wrong way.
Yeah.
Reach RV, can we go ahead at 0.2?
Just wait until it gets
Yeah.
track.
Okay, that's it, thanks.
So now there are so many people in here. Temperature is going up.
Yeah,
not
the
me.
shift of the pilots. So meaning that it's lunchtime.
Oh right, yeah. Yes, no, but take them out. Yeah, I'm just coming back. Yeah.
So how long until 17 minutes?
Yes.
Still change the tape.
Are you hungry?
A little bit, yeah.
So we are around lunchtime on the boat and around shift time for very many of the crew. Both the ROV pilot crew and quite a lot of the crew on the boat as well. Shifts around the meals.
How are you doing?
Yes.
Just ahead, just we've got up here.
We ask ahead what he's doing. Northeast.
Then we can left. Go left.
We ask to do that.
Well, I'll
Just trying to get again to the transept.
Yeah, but we are just turning around, and it's easier to turn around if you're a little bit up, especially when we're changing everybody flying as well. And it's good to be either sitting on the
Can we
ground
have
or
a change of bearing to 180 at 0.2, please?
So this is the reason why you're seeing this blue screen now.
We're making north, we're going, isn't it?
yeah, we're going north.
So why is it
So face now.
Yeah, because we went down, we should go up with the other. We did a loop there, and then just coming down, and then go up the first, and now
Back up,
north.
that was the red job here at all. Yeah. I don't look at eye, just go back at eye.
Actual left, yeah.
Oh no, the lateral left, go back to forwards.
I can't see.
Yeah.
Look at the range of the solar there. See what that
on
is.
the shot,
I think we confused them a little bit with all the turning around.
Yeah. All these
right.
Searching for the
Just push forward.
Yeah.
yeah, best thing would be supposed to have been to go to continue the transit and then get proper response.
That way.
Yeah.
You're pushing forward.
Yeah, just
You're going.
so
From southwest, like to bring your reading. Your reading on clockwise here.
Clockwise we'll go. Because we came counterclockwise to go left to look for those.
90 degrees.
Yeah.
Ridge is kind of here, we're way off the ridge here.
okay.
West there's keep an eye, see what changes. Just forward, you know.
Yeah, just forward, yeah.
I see you're still going northwest there, so bring it around there, 45.
So we are 40 minutes away from the ticket now.
Yeah. We took a little bit up and then we will go down again and find the seafloor for you all. No worries.
There you go.
The transit will continue.
Yeah. Okay.
So that's a ridge here.
That's a 50 meters range. So bring that down as you go along.
Keep looking up, it's freezes again.
Yeah. Now I see the seabed.
Yay! Back to the seabed. We like the seabed the best. Yeah. But now we can see all of this
Yeah.
marine snow in the water.
There's a wall.
Yeah.
Yes.
But
Did it continuously move or for a distance?
No.
Continuous.
Continuous, okay.
yeah, we're coming right up until this wall.
That's 0.2.
So how do we end up to the southwest?
Because we go and when we pick up, let's go left to look for to come round, to go backwards,
So
to look
now
for
it's 10 meters
sponges.
north.
Okay.
That's where we land, we're
Yeah.
pick up, so go
now
and
we
behind
can
us and go back.
Going up.
Okay.
Okay, so yeah.
Yeah.
Yeah, that was us. That was our fault.
That's where we
But look at this. We have
it's off to the left.
the same wall
Yeah, similar as
fauna again.
previous one.
It's a very interesting combination of everything.
But I'm going what he said.
Latest.
It goes on.
Yeah, thanks.
Asking me, and I've given the fucking answer. Right, no fucking ask.
Alright, that's alright.
And our next shift is coming in as well. So I hope they have already had lunch.
Yes.
Yes.
Noodles.
Yay!
It's okay, that's good.
Comfort food.
I'm not sure that.
So we have done a lot of we've done the push course and the whiskey and one coffee mug. And did you see we tried to get the Slaughtersaurus? It got away. Free. And then we also this we didn't get. This was a try to get the small soft corals.
Yeah.
The ones that are on the edges of everything, but they're
Spend
always
some
on the
time.
edges of big things. So Sergio wanted us to just pick up the mountain, and that didn't work.
Uh-huh.
You always
What
wanted
was
that.
something
That was
lying?
interesting.
On a bubble.
But that's the jelly.
Oh, yeah.
That was a nice jelly.
So the idea, guys, was to once on the top try to get one of these demo sponge. Piece of demo sponge.
Yeah,
There's not that many.
Which demo sponge? Demo sponge is
Yeah, like not exactly
any demo sponge.
Yeah,
because yeah, it's
he
for
wants-
combining the mud. I mean the sediment, push core, and the skin with some species, with at least one species that filters a lot. Because exactly, and it's they tend not to.
But then shouldn't we take geodia?
Geodia, for sure not, because it's a high prevalence species. So I think one of these fun-shaped ones may have, yeah. We saw, yeah, we saw
So
some
videotape change in 10 minutes.
many.
Many options. We saw
There's no
Lysodontorics would be
Lysodontorics.
nice.
So if you find one Lysodontoric, just
That's, oh, zoom in on that. That's squid.
Let's go with something.
oh wow.
Wait. Could we stop?
Yeah. With a fish.
With the fish?
Can you go? Can you try to chase this
No, don't go.
a little bit? Get closer.
It's a squid with the fish.
Oh yeah.
Try not to move the camera, then I'll go.
Oh, sorry. Look at that. Oh.
Yeah, he is hungry. Yeah. Not that way. That way.
We need the whale eating the squid.
But that was nice.
Yeah, alright, sound. Right.
Can we take the lasers off?
Yeah.
Lasers off.
There you go. Thrust and wash.
It's getting out from the still there.
I just wonder how he's going to manage to eat that whole thing. He's going to be full for a week.
Was that carrying eggs, you think?
That is eating, catching a fish.
Lovely.
So it was a squid having caught a fish.
You're up there.
Yeah.
Hunter.
Nice one.
Yes.
Yeah. So I'm following the trail.
Hold on. 2 more minutes.
8.
Where's it going?
Is that one of these fun shape?
No.
Yeah.
Is
Is
it?
this fun shape?
Yes.
Can you stop here, please?
Yeah?
Because we're gonna grab one of these.
One to the right at least, one to the left.
That one, sure.
So finally, Sergi, right before he's leaving for lunch, found his fan-shaped sponge that does not have a lot of bacteria and filters like crazy, and will be his
That is the one.
active natural
After 20 minutes.
filter for the eDNA.
Yeah.
And it's good because then we will have a happy Sergi going back to the lab.
Yeah.
And we aim for
We're going
happiness
down-
in
yeah,
all
we're
our
going- we're
people
going,
here.
going down slow, so the back of the RV is hitting the cliff.
Okay.
If I go forward, I'll go over it. So
Uh-huh.
this is,
Can you land it here?
no, because it's going down that way. I can swing around
Yeah.
and face up.
Yep.
Yeah.
Like there.
Okay.
Should be.
And now he's
yeah,
redirecting
now I see that.
Yeah.
the entire RV to go around to get his fan-shaped sponge without much bacterial
Yeah.
action.
And have you had a good catch of Lysodontorics?
Not really. We didn't do
We
many.
haven't done any Lysodontorics.
So we
But
were
we've had some before.
four.
That'd be fine, but the one that we took
If
on the
you
biggest
if you take a few more, that would be good. The thing is that we don't see many in this plateau area, so they were more like not up in the wall, but just a medium depth, probably.
Okay.
And
You think you
Yes.
can reach that one? Probably
Should be able to grab or slurp.
Uh,
Grab.
and see the beautiful spider right next to it.
So I'm going up here then. I'm going up here.
And then we found out that
Yeah.
these little orange dots in the foreground are also sponges, right? I think it's pretty.
Mm-hmm.
Well, I mean, just aesthetically pretty.
Yes, okay. Yeah, swim around for yeah.
So it's 5 minutes.
Yeah.
5 minutes until tape change.
Oh yeah.
There you go.
They're ready. The one behind
So
you.
one on the back.
One behind.
Yeah.
Up it. Is it?
Yep.
Yes.
Can you zoom in before because I'm not so sure about the idea.
And we need the lasers on.
Huh?
So you have a.
That's one is to find the spots. Make sure it's not an insect in it.
You think it's an exactly?
Yesterday we collected insect in it. Similar to that one. You see the skin? You see the skin?
Uh-huh.
I'm not saying it is, but it's better always to zoom in before collecting some.
Yeah.
Oh, there's a
You see the
little
skin?
jelly.
Not really the spongy.
Well, we got a quite large part of that sponge
So
now.
that is it.
Where am I going with this?
Yeah.
Yes, please. We can put it in
Firebox.
the which is easiest for you. There's space in both.
Have to be the port.
To the right.
Okay, you want to have it in the
The one on
port?
the right there.
In the port, both in the back are free.
But this one has the mug.
Yeah. No, but that's in the front. So the
Yeah,
front.
That's gonna be a little bit.
Yeah.
It was broken, right? Already? Had like
Can we
a.
stop the movement for now, please?
You will see in the images. So this part. Está roto aquí. You see? It's not a complete fan. It was just broken.
That reminds me of the one that they collected in shoes. You know, the one that is right.
It could be. But I think this shape of this has more power of filtering, right?
No, you want
Potential.
the more spongy.
Yeah. There's no other than that, right?
Yeah, we look for one.
If you find this lyrics for sure, it's,
What are you doing to score here?
we did everything already. All the push cores and these in bottles.
So it's just about
And one mug.
So you can do one more mug and you can do little Endorix, and there is quite a lot of space still. We have not slurped anything, so there's 2 slurps left for you.
How far are you in the concept?
Not very far.
which mark is left.
The front left. Okay, so it goes in 4 instead.
So it's gonna be EV4.
Yes.
Thank you so much, guys.
Okay, 1 minute until tape change.
And 1 minute devoted to this pycnogonid.
Yes, 1 minute devoted to the beautiful pycnogonid.
Ooh, look at this beauty.
Hey. And to the
Okay. Oh, carnivorous sponges.
carnivorous
Oh,
sponges on the side. Fancy. And then we were agreeing that these were tiny anemone.
Mm-hmm.
The little
with
brown,
tentacles in.
yeah, the little brown orange thingies are anemone. But these things up here, they are sponges sitting on the orange.
Or anemone. Could be, right?
Yeah. You
Yeah,
think they're
anemone with,
anemone still, but sitting on the,
Yeah, because there's
they're sitting on the sponges, right?
Yes.
It's a beautiful specimen.
Now it's coming. Okay, it's going to come and check
On
out
the
the.
run.
Okay, please change the tape.
1, 2, 3.
Stop recording. Thank you.
Yep. Okay, guys.
I have an empty one for you.
Okay. Thank you.
Beautiful shot.
Ready to start?
Yes.
1, 2, 3.
Start recording.
Anemone.
Yeah. Okay, then Anna-Helene and Sergey are giving over the microphone to Franzi and Julio.
Yeah, let's go for lunch.
So the next group will take over then. Yes. Okay.
Thank you so much.
Thank you guys.
Okay. Yeah, you're welcome. Yeah.
Hello, hello. Julio and Franzi are back again.
Belly full.
Yeah. Exactly. Some nice soup. Okay, I give this to you.
Yes.
Thank you.
Right, what am I doing? Am I going just straight up?
Must be
Back on the same.
So cool, these spiders.
Yeah.
Speak now.
I like the
Their
spiders.
mouth. But these ones, a little bit more.
Yeah, maybe it's a little bit more.
Their mouthpiece is really cool. This elongated trunk.
Will you put the laser on? Thank you.
Okay, let's see what else we can discover here at the unnamed seamount. Many more of the shrimp friends.
Yeah.
So one mark,
Yeah.
some is under its next. That's
Mhm.
it, right?
Yep. Ooh, there are a lot of sponges coming up.
Yeah. How about stopping in this
Oh.
area?
Hey, this grows very interestingly.
It's tricky.
turn.
Looks like foam.
And the other one. Quick make it turn.
I'd like to check those big ones.
Yeah, what are they? These ones?
Yeah, I think that is the same that they fish over there. They collect
yeah. Without
on the first dive.
I'm just going to come up. I'm just going to come up a little bit.
Yeah, no
So I
problem,
can
no
turn.
problem, no problem. Sure.
Okay, because it's going to be a big turnaround, isn't it?
Don't do it if it's
Yeah, it's not so easy navigating on this terrain.
I'll put a big turn in the
So our pilots have to figure it out a little bit.
He's got to get that down, otherwise we could twist into the orbit.
Is it okay?
So we're making a bit of a turnaround now. I already lost my directions.
Yeah, we are spinning.
Sorry. I just need this.
Ooh, okay, now we have a nice view.
Click yours.
Do you think it's the one that we just sampled, or Sergi and Anna sampled?
It's a bit hard to say.
It's a really cool structure. Is it even, would you still call this fan-shaped? Yeah, right?
Yeah? Yeah. Well, I mean, those are just names, you know, so sometimes the shape is not that clear.
No.
It's a transition between, it's something in the middle, but yeah. It's a fun shape. Lamellar also, sometimes fun shape. It's more when you have a clear center at the basis.
Yeah.
This is more like lamella because you don't have a one point where the fibers converge, but many attachment points. See what I mean? It's like a wall.
Yeah.
So, but because they are sponges, they change the shape so much. That it's always just to have a reference. But yeah, you could call that different shape. I would say that this is the same that they just collect.
Yeah, looks
So,
from the inside structure also.
Yeah, I will not collect this one,
No,
but I would like to have a nice look at it.
Yeah.
So to see the
community around.
to see
how
the
it
morphology
grows.
of
Yeah.
it, and maybe we should call it a little piece. The thing is that if it's a new species, you want to
That's
have
good to
a
have
nice
more.
view of the one that you are going to sequence and deposit. I'm just afraid I don't want to break it. Seems like it's a little bit.
Yeah, it looked as if it's very.
It's not that easy to break, you know, because.
Yeah, to break a small piece off. You'd rather get the whole thing then.
So, I don't know.
Do you have an idea which family?
No,
It is?
no, not
No.
yet. Not yet.
It's really cool, also vein-like structures, not only the pores, but also these,
Yeah, well,
veins.
those are the fibers that give the structure and the like the internal architecture of the whole body. So.
And it's growing on top of geodia?
It's rock.
Okay,
This is
yeah,
rock.
yeah. Not the furry geodia sponge.
I'm not sure if Paco is over there, but
I think he's busy with another school.
okay. fancy that if we try this with the hand. I just need a piece. I don't need.
Yeah, might be a bit tricky to get it off, but you can try.
How big is that?
Where the lasers 10. It's over a meter, 1.5 meters maybe.
Could we turn the laser off for a second? Thank you so much. Another one. Yeah. This is nice also in the
In the back.
background.
Yeah. Some small corals and sponges and anemones. Do you think maybe here at this junction it's easier to break it off? I mean, then it's a...
This is very big, huh? I don't need that much.
Yeah.
I don't need that much actually, but yeah, we can try. I mean,
What's this? Is it a carnivores?
that's coral, I think. It's kind of.
The polyps, right? Yeah.
Yeah, those are. That's coral. Some polyps. Let me just check that rock over there. It's just kind of beautiful. Have this community of anemones.
And worms? What, like tubes of the worms?
Sure.
Oh, worm guy Sergi is not there anymore.
So I would try to pick a piece of the large one. Even here.
I would think it's, yeah,
It's
I mean, this
easier.
is much bigger, but I would suspect that that's easier to break, even if the piece is bigger than the one that we want, because it seems more loose.
Okay.
But,
Okay, let's try that one.
yeah.
Could we try to take this piece? Big piece on the left? Yes.
If it's a smaller piece, even better, but we suspect it's maybe it's breaking here. But if you manage to break even a smaller piece, even better. But might be hard to break it off
okay.
because of its structure.
It's kind of a fibrous texture. So
That's why that's what Rory put there, was that they better be.
Where would you like? Might not be able to fit in the starboard side.
So starboard is not available.
Might have to reposition. Yeah.
But this is in there, so we could put it there.
Yeah, so
Okay.
port back left is empty. Is available.
Yeah, it's pretty big to get in.
It's too big.
See what comes.
Might be able to get it.
I think we should, yeah. They did zoom?
Zoom in.
Yeah, tilt up. This is fairly hard.
Maybe if you just
Yeah,
Snap.
I'm just touching it, so it's also fibers.
Yeah,
Yeah, that's
that's
the one.
already. Yeah. Very fluffy. Perfect. Yeah.
Is that? Yeah.
Really nice.
Do you want to report?
We try port, yeah.
It's a reminder.
Yeah.
Do we slurp this up now?
Slurp, then everything will break, I believe, because it's too
But if we put it,
fibrous.
and if we put it
You
in,
want to slurp the piece, a little piece? Yeah, that's fine. That's
because
fine, no
otherwise it
problem.
might flow off, no?
Yeah, that's fine. Yeah.
Okay, close the drawer and slurp this piece.
Good one. Yeah.
Yeah, I thought you wanted to screw it.
Yeah, so,
So this is cast 11, right? Yes.
before you do that hand drink, you
Yes.
get the start tank and you're ready.
Do you?
Slurp.
Oh, 4.
4.
Yeah.
It's okay.
It's okay to go with 4, yeah? Okay, so zoom in a little.
Sorry.
Let me know when you're in position, I'll come to you.
Sounds good.
Yeah.
Very great.
The zoom there, Julio.
That?
That's fine, yeah. Where are we? Just no constant. You good there? Yeah, going in. Okay. So suction on that?
Yeah.
You're on, yeah?
Yeah.
Okay. I'm gonna open the jaws now.
Perfect. Nice.
Okay,
Thank you.
number
Well, that was some nice sampling here.
4. That's it for sampling, yeah?
Yes.
Okay.
Thank you.
You wanna follow?
I can follow the line.
Yeah, 1 second, I'll get the arms away. So get your hand ready first.
So we collected a piece of this sponge that we don't know yet what it is, and to find out later on what spicules it has, and in general what more we can find out. Will you analyze the molecular genetics as well of this?
Yeah. DNA. DNA and morphology. So hopefully we can put a name on it.
Yep.
It's important because it's building an habitat. So when they are that big, we really want to know what they are.
Yeah. Yeah, they're super big.
Should I move the camera?
No, you're okay. Oh yeah, you can square
We
up
don't
your
have a
camera.
lizard and Dorix here, right?
Put your camera in the middle. Okay, Rob.
I just wondered if there was a lizard and Dorix here by chance, but I don't see any. So
No, I haven't seen any. So we need one mark, right?
yeah, we want one more mug with sediment. And we have another slurp chamber. And a lot of space on the starboard side. For listener dogs.
How far we are on the transit?
Apparently not very far. And try to understand.
Is it going up?
Understand.
Is it going up, or is the summit? Is it going up, or is the summit? It's going up at the beginning and then summit. Yeah. So yeah, maybe here at the end. So this is 1,451 meters.
1,051.
451 meters.
The flow, using changing
Yeah, so maybe we're here.
Around here.
Or we are here again. But no, it doesn't look like it.
Yeah, maybe.
Okay, guys, if you're ready to move.
Okay. So I'm gonna put a half turn in.
Yes, we're looking in our smart folder here, where we have all our different dives nicely laid out, with a map and with a profile of the different depth. And we're now on the third transect for this site. We're at the unnamed seamount, far up north, along Knipovich Ridge actually. We're not on Mons Ridge anymore.
Oh, okay, yeah, that's a good point.
Yeah.
Didn't thought about that.
Yeah, I mean, neither somehow. We just, had another dive tonight, or last night, at this site.
Flat there, those rocks.
Yeah,
Probably. Yes, that interesting. So
where
rocks
we
are
explored
super
another
flat.
side of this
The
seamount.
rocks were super flat.
Also, a slope. We're climbing up a slope, and now we're climbing up another slope. And yeah, it's really interesting to see the communities here in comparison to what we've seen before further south. And similar depths though. And in general to explore a site that has not been visited before or not been explored and studied before. For sure, it seems that there's a lot of biomass in general, biodiversity, and it also seems to be a very productive area. We've seen from small organisms up to big ones that there's a habitat built here, a lot of shrimp and filter feeding organisms, then some squid we've already seen, also in action eating, and how this whole food chain builds up here. Yeah.
Could we turn the laser on?
Yes.
Thank you.
Now it's Mallorca time here. It's t-shirt weather.
Mallorca time.
And I'm all dressed up here.
Are you cold?
No,
I think it's okay now.
Yeah. But I don't feel like sitting in a t-shirt.
It's not Mallorca weather at all.
Julio is from Mallorca, that's why
Yeah,
we talk about Mallorca weather.
a bit warmer there.
Okay?
A bit warmer there.
Ah, yeah.
Un poco más caluroso.
It seems to be more even now. Or going down even, I mean.
So, could we go to the port?
Yeah.
Just
towards
follow
the edge.
this
Yeah.
crest.
Are those sponges Rosalide?
That seems to me a mix of Rosalide and what we just collect.
Ah, yeah.
But I'm not sure actually. See the large one on the rock is what we just collect. Almost sure. Then you have some Rosalide.
Yeah.
You have some carnivorous sponge. So somebody is feeding off those sponges. You see the holes on those sponges?
Yep. Maybe
That
we had an hungry
seems like
system.
spongivore.
Spongivore.
Yeah. And you see the bald rock is completely covered. Well, completely covered. It's covered by those anemones.
And what edge is here?
That's the rock.
Yeah, but it seems like these outlines of it. Is it just sediment on it? That seem to have like an encrusting outline.
All these large little trees are also sponges. Those are carnivorous sponges. Different species you can see here. This one that is more branching, and another one that's more like one single stem.
And this is the more branching one is the Asbestopluma, and the other one.
I'm not
like
sure.
suppose
It could be
potentially.
Asbestopluma as well. Could be-
Acostatus too.
Of the second one?
No,
as well?
no, of these ones.
No, this one, yeah,
Yeah.
for sure.
This is nice.
Just stay with them because something going on.
All those in here in the back.
Yeah, that's the same
The
one
fan.
that we just collected.
This is 50 meters, sir. Just wait. Okay. Yeah.
What's there in the back? Is it a fish?
It's about that position.
Or is it a squid? It's a squid.
Yeah, just looking off.
Yeah.
It's more like a tenaphora to me.
Ah, oh yeah, I was. Yes, I had the wrong. I thought it was much further away. I didn't have the 3D comparison. It was like a optical illusion. Yeah.
We have a movement bearing 005 at 0.2 knots. Please continue with me.
5 at 0.2.
Thank you.
There's a skate over there to the right.
Where?
There.
Ah, there in the back. Ah, yeah. Nice. We haven't seen skates yet on this dive, I think.
Or even on this area.
Yeah. That makes sense, since it's very productive. Like,
I'm not sure.
no? Yeah. Let's see.
I'm not sure.
Oh, it.
Yeah, there she is. Looks like a female to me.
Yeah. No claspers visible along the tail.
Yeah, I got it. Yeah.
We're here at this corner.
I guess we are around here.
Not here.
Well,
no.
maybe.
Yeah. And these little ones are all the anemones, right? The
Yeah, I would say so.
orange,
Otherwise,
dark
species
red.
of anemones, of
Oh, the
corals.
skate is flat. Can you zoom out? Or disturbed its nap. Her nap.
Could we stop for 1 second? See if we can get the sky the other way.
Yeah. Okay.
Yeah, that's fine. Thank you.
Let's see if we also see some eggs or so.
Yeah. We haven't seen any so far. Yeah.
No.
Okay, keep moving.
Okay.
Funny marks here. Somebody
Yeah.
wrote his name like an A to the right, T and A. Ta.
Who was here? But yeah, like
Signature.
interesting. Why are the marks here?
An alien.
Oh, alien. Yeah. Let's come up with some conspiracy theories. They are wondering as well. The deep sea aliens. But yeah, to our knowledge, there hasn't been a research expedition here. Maybe also,
That was maybe the skate, I think.
and it just looked as if it was man-made, human-made.
Don't you think that it was the skate? The skate marks?
Could be, yeah.
Yeah, laying there and chilling a little bit,
Yeah, and then
moving,
doing it.
shit in the right
Yeah.
posture. More marks.
We have a lot of the little. Are those also the anemones? The little dots we can see, or are those sponges?
Those are sponges, I think. Well, probably a few things over there, but
Yeah.
that looks more like a sponge to me.
Yeah, yeah.
I don't see tentacles or.
Sleepy anemones.
It's like
Yeah, like a path.
A little path.
Yeah. We're on a hike. Where do we go, left or right? Or we just stray from the path? Yeah.
You want to follow the path?
No, just into the unknown. We follow our transect into the unknown.
Sorry?
Shall we follow this mark?
There are a lot of marks. think this bottom are foraminifera? It looks from the structure a bit like something.
We've been sieving the samples this morning, and there was this gravels with a little bit of mud.
Yeah, that's true.
Yeah. Yes.
There are a lot of little stones.
Yeah.
Reach RV. Can we increase the speed to 0.3, please? Thank you.
Polymastia there to the left. And this is a cladorhiza with a little tree. That's cladorhiza. See another polymastia.
Delays, you think? Or it's too difficult to tell on the species level from this distance?
It looks like the air, but it's not that clear.
Do you think we should take a sediment sample soon here in this area?
Let's do it here.
Yeah.
Okay, to stop here, somewhere around to do the mark.
Yeah.
Okay.
Thank you.
Maybe here in the corner or so?
Is there a specific spot?
No. Just to get the sediments, not the rocks.
Yeah.
So maybe a little bit more port side.
Yeah. Could do that.
A little bit. Ambition.
Yeah,
About
un
to say.
poco.
Ambition.
Un poco.
Yeah. It's not it.
Oh, shrimp. Oh, do I see a Lysidendorix in the distance?
Yes, we will collect now. Thank you, Francine. Let's plot.
Do you want us to? Yeah, 50 meters.
Let's collect the mark first, right?
Yeah.
Here. or
Yeah.
where we-
Reach RV. Can we stop the movement for now, please?
Here in the brighter part, maybe it's good.
Yeah.
Thank you.
We will have some.
Well, I'm not
Some
sure.
pebbles.
I'm not sure if they want. Maybe this looks like foraminifera actually.
Yeah.
So maybe
And I
they-
don't think they just want pure sediment.
They probably want a mix of everything, right? So they're looking for things inside that more diverse. Don't think so.
I thought they just want an area where it's not covered
Okay.
by anything.
Then yeah.
With they, we're talking about
Done? Which of the coffee mugs is done?
the right one is done, right? So the left one is, I think, still free.
From left on port.
Check. Yeah. Mhm.
If you zoom in a little bit, just follow the jaws. Which one are you going for?
So we have a team of scientists that are
So you want me
interested
to zoom
in
here? Or-
Yeah.
in
Keep that
different
on the
faunal
jaws.
communities here, and
The boats.
we are collecting sediment
That way.
samples
You see it
that we
moving.
will sieve afterwards to see what's living sediment. And how it is composed.
Not.
And we already, or the other shift, Anna-Helena and Sergi, they already collected another mug of sediment, as you might have seen. And we're gonna do the second one here. Oh, there's also a snail just above, but yeah, you zoom in on the important part right now to give also the pilot a little bit of support for knowing where the arm is at the moment and how it can grab the mug.
Oh yeah.
have a specific area? Or
Where it's more clean, for instance, I can show you here. You see?
Okay, that's fine.
If it's okay, yeah?
Oh, yeah.
Yeah.
Then we find other things.
That's where you are. It's a white area in the red. Swing it to your right. So tight.
Yeah.
Right.
But the trace in the middle. That's what you can say. Okay. All yours.
That's it.
Wow, that's good. Nice one. Beautiful, thank you. How do we want to go about with the Lisodendorics?
Oh yeah, the design lights, I forgot
How
about
about this, mesmerized by this sampling skills.
There is a sponge we would like to collect.
We have another sample we would like to take, and the distance in the top left corner, there's on the rock a little Lisodendorics, or even two sponges. And the sponge experts are doing connectivity studies on them, so we collect multiple ones of them. If you've been following our dive streams for some time, you've probably seen us collecting quite a lot of them yet, and know everything about our objective here. But in case you just tuned in, this is a really cool sponge called Lisodendorics complicata, and it grows basically everywhere. So we have it here in the different depths that we were, from like 600 meters until almost 3,000 meters. It was just always there.
Just one shade that is actually, yeah. It is growing on. It's growing on Angelia parva, and it has an amphipod. If we
It's
could
a
collect
big
the amphipod, so that would be slurp. Yeah. So here the problem is that this is attached to this.
Yes.
So everything will come.
We won't,
That
like
is
the
not-
cylinders might come
It's
loose,
too big for the slurp gun, maybe.
but you
Can
don't
we
think we
pick
can
it with
slurp it?
the starboard minute and suck it then?
Yes. That would be perfect.
But the amphipod might escape.
You don't want the
The amphipod will escape, so
What's going on?
I think the
This is polyline, right?
because
I mean, we can slurp the amphipod first, and then
It looks like an interesting amphipod to me, but who am I to tell?
So shall we slurp it first?
what's
Francie?
the one you want to start first?
The little amphipod
The little
The red
trap.
one?
Yeah,
orangey
amphipod.
thing?
Yeah,
Yeah.
the orangey one.
on chamber 5.
Is that an amphipod or a polygid?
I don't know, sorry.
That's a polygid.
Whatever.
Definitely. That's a scale worm.
So we don't want that.
Oh, yeah.
Oh yeah?
No.
Chamber 5.
But it,
Are you sure about that, Sergi?
Ah,
Yeah,
yeah, I'm
it's
pretty
look-
sure. I work with the polygons, yeah.
It has legs, huh?
Yeah?
Yeah. I'm not.
Yeah.
I don't think it is,
That's a big one.
but it has.
Segmented, right?
yeah, it has
You see
segments.
segments.
Yeah,
And you see like a kind of a...
Take a look.
Yeah.
and
Yeah,
big
go on.
eyes and
You're right.
some mouthpieces.
But what is weird is the legs.
Yeah,
See?
small
Legs.
Ah, no.
cockroaches.
Yeah, right.
See you later, Sandra.
You're slurping it.
Who's going first?
Yeah.
What are we going first?
Yeah, I thought it was like a
We're
polygon
going to
was
slurp first.
coming here like crazy.
But you don't think that if we slurp this would come off? The geodia?
The Geodia will come.
No, I mean the Lysodendrix will come off,
That's I don't know.
because that way we had.
Yeah, you should slurp it.
The thing is that it's attached to the Geodias.
Yeah.
And they will be
If you
messy.
take a lever,
Well, that's on them.
Yeah.
I mean, they will start by slurping the amphipod or
The amphipod,
whatever.
yeah.
Maybe piece of
There's plenty of
Uh,
amphipod here, right?
So,
plenty of everything.
Yeah.
I explained the situation.
Okay.
We can slurp the amphipod. If we can slurp the sponge without taking the other two large sponges, that's okay.
Yeah.
This sponge is a little bit brittle, so it may break if you slurp from
Yeah.
certain distance.
Yeah.
If you get too close, the other ones will come.
Yeah.
So maybe we can try to slurp.
So you
from-
want us to make it impossible?
Make it possible.
Yeah.
Make the
Mission
impossible.
impossible.
Yeah, make it.
Make the impossible.
Okay, so what I'm thinking of is bringing the starboard arm around the white sponge, and we slurp inside the jaws, so we hold the sponge down.
That's perfect. That makes sense.
Isn't that sound so easy? Okay, let's try and make this happen.
We have an amazing ROP team here, making the impossible possible and always coming up with amazing solutions for the different samples we would like to take. So yeah, we're really blessed here on our expedition with this team of ROV specialists.
Okay.
Porque es que ya es que voy con
Sí. Bueno, no sé.
y que él intenta tocar esto. ¿Cómo es?
¿Cómo es? Lo hemos visto, sí. Sí, sí. No, okay. That simplifies everything.
Staying there.
Is it a,
This is my house.
Zoom in for me.
not so much?
Sorry.
This, yeah. It's like a ping pong kind of a sponge.
It's kind of a sponge.
And the ping pong balls. There are these ping pong carnivorous tree sponges.
No, it's not.
That's
no,
not
no,
that.
no,
No, they look different.
Right,
Yeah.
no. Those are big.
Yeah. I didn't see them yet.
I gonna pay attention to this because it's really... So they want the orange tips that are laying.
Yeah.
Okay. So that's the reason to keep.
Yeah.
And frozen there, so
Yeah, because you want to be preserved in
you
the
might get a
liquid.
little bit of the darker brown sponge.
It's okay. No problem. Shall I come with Andrea?
No, you can leave it there. That's fine. Thank you.
Sorry? Yeah.
That's fine.
Come on, listen and
Okay, let's go back and touch her off.
I'll have.
Yeah, that's not.
Big paper falls out.
Yeah.
Perfect.
Oh, nice.
It's okay.
Stop.
It didn't go through. Second, it didn't go through Sterling's and the nozzle.
Stop. Just
Ah, yeah.
time.
No, just look at the nozzle and fall out and shake it out.
Come out, little Georgia.
There you go. Come.
So we're trying to sample just what's necessary, but it's sometimes not so easy to separate everything from each other. So yeah, we're trying to get at least the Geodia out again, if it's still stuck in the slurp house.
Can that sponge go into another box?
Yeah, no problem.
So we can do that, Rob, and then just suck the whole lot into another box if it's not coming.
Yeah. So that probably gets stuck.
Oh no, then you go through.
Okay.
Oh no, you're gonna move the box. What number would we put that other sponge into?
You want to have a
Two
look?
biobots.
Oh, there's the one.
So it has to be.
The number we have, the number 5 already has one more, please. This is slurp, and here is, what is the
So, slurp or biobots?
Slurp.
Slurp 4.
Four.
Yeah.
Alright.
And then we have the Geodia and
Okay.
the Lysilendorix all in that one.
There was no Lysilendorix on 4. Now there will, now there will
Yeah.
be a...
Okay, it's in 4.
Perfect, thank you. Now, thank you.
Okay.
Hi guys.
There was no visual storage.
No.
Huh? Now there is a two geodias and one visual storage.
And nothing got in number 5.
I'm not sure. Maybe the first geodia.
Let's check.
But we will know.
But let's check already if they know. Did anything go in slurp 5?
Yeah, we put the white bit into slurp 5.
And the first other one.
Yeah.
Yeah. Good. Thanks.
Okay. So I'm good.
Oh, that was 13.
No, ja mam tutaj.
I have too many papers here.
From my side, I'm okay. Anytime you.
All right, good.
Where you go?
What?
No, fancy. Where you go?
Yes.
Yeah, shall I help you? Something.
Yes, it is.
Sorry.
I have it
That-
completely under control here. No, there was just the papers that were in other orders. But yeah, so we have first part of this cast on here and the
Geodia,
other.
and then another geodia with the fan-shaped sponge pieces.
Yeah.
That's it. Sponge soup, 2 scoops there, and those are free. Do you want
Yep.
any product?
No.
Plenty of space.
Yes. Yeah. The shape looks different. No?
Bridge RV.
RV bridge.
Can we resume the movement at 0.2, please?
005 and 0.2.
Thank you.
Okay, we're continuing our exploration here. And after we've climbed a very steep wall slope of the seamount, we've arrived at a more flat area, more sedimented area, and it seems to be covered with foraminifera, at least at some parts.
Yes. Yes.
Sorry, you wanted to say something?
No, just that I think that they are looking for those orange tubes. I'm not sure what it is, but
Which orange tubes?
You remember there were a couple of orange tubes before that we were
I
wondering about.
thought like those were broken off pieces of the asbestopluma sponge, of the carnivorous sponge, because at its stem it also had more orange shade.
Some are, yes, I
Yeah.
think, but not all of them.
Yeah. In the distance there's a fan-shaped one, right?
Yes.
Yeah.
What?
A fan-shaped sponge. Yeah. And a carny.
Yeah, I was the pluma.
Still a lot of shrimp hanging around.
See,
On the
okay.
bottom.
I can get this one. No.
Ooh, no, I didn't want to go in the plume.
Very white. So I'm not sure what it is.
What is?
The sponge, but I guess that is the same that we
Mm.
collect.
Do you already have ideas how you would name the seamount?
Uh-huh. Are we talking about that?
Because it still has no name, it's a blank one.
Seamount, the anemone seamount.
Yeah, I was also thinking of something like that. Because we saw so many nice anemones before. Is there a fish?
It's a skate.
Is it? No.
It's a fish. It's a fish. It's a fish.
I'm not gonna touch it.
Yeah, eel pout maybe?
I'm not sure. Big one.
Hmm.
So it seems that the summit is more sedimentary.
Yeah.
I think it was the same this morning, right? A very nice slope and then
Yeah.
more muddy gravel on the summit.
Yeah, and in comparison to the earlier dive that we had at this mount, we are now at in a more southern region, and the
More
dive
southern?
before, we are more in the south now compared to the dive we've had before.
And the depth?
In comparison to the earlier one,
Yeah.
so the earlier ones were between 1.2 and 1.5,000 meters depth. And this one is now 1.4 to 1.6, so a little bit deeper.
A little bit deeper.
Yeah. Further south and a little bit
It's a
deeper.
crinoid there. I think it was a crinoid covered
Oh.
with anemones.
Oh, and have we talked yet about that we are now on the eastern, that this seamount is on the eastern side in comparison to the western sides that we had in our previous dive, so we are also interested in comparing the seamounts east of the ridge to the ones on the western side, and also taking then the flows of currents into account and what they might do the community structure.
And the thing is that historically the western part has been more well studied by the Norwegians, while not much has been done in the eastern part. Probably nothing, maybe. But I don't want to say that because I don't know. But in terms of publications, you have some nice studies on the shoes. I think that there are some preliminary studies on the Void, very preliminary, and on the Mons Treasure as well, but not here. When we collect the biggest sponge, that was...
We're going...
It's like we
four, six.
yeah.
We were already like 10 meters shallower, but yeah, it didn't differ much. Let's check the depth profile. Yeah, there's a part I think where maybe at it now where it goes, the slope is slightly going a bit down, maybe 20 meters in total or so, and then it goes up again on a little peak, and then it's
That
fairly
peaks-
flat again.
That peak looks nice.
We're looking forward to the peak.
Maybe where the direction changes.
Yep.
Do you know how much is the resolution that we have in this area?
30 something, so fairly good.
35.
Yeah, 35 somehow, more or less. Yeah, so while we were planning these dives, we had different different resolutions of the seabed already. What's going on?
Okay.
Oh no, I think our pilot bumped her leg or has problems with the chair. Happens to the best. Yeah, so we had a different resolution, so the accurateness of the seafloor data that we had before was different to each other depending on the sites that we visited.
Is it
Sometimes
going down?
was more clear and sometimes
This
more
is up, right?
coarse.
And then it will be turned into port.
And in the back we have one of these bigger fish again.
Still going up.
Similar to the one we've seen before. Where's the fish sheet? The fish sheet is not there. But it looks like a big eel pout actually. But it just has a very big back.
They're turning up.
There are more of them. Oh, this one has a big mouth. Looks like it's like these big, the fish, demersal fish sitting on the ground and just having a big mouth that they can snap open and close again.
How big is that fish? I think.
Huge. Yeah. Something like that. Oh.
There's plenty of them, no?
Yeah. Oh, is it the one that just swam off?
Maybe. So we're trying to get the proper direction because we are at the very top.
Ah, yeah.
Bridge RB.
Okay, bridge.
Can we change the bearing to 350, please?
50.
Thank you.
Some Ascidian carnivore sponges and geodia and soft coral on top as well.
Yes. And some polymastia. That's good.
More fish. And are there some of the fan shape in between, or are they all Rosalide, no?
But
Mostly, at least.
so that one, yeah. This one. You see? Can we stop it?
oh.
Well, maybe too late. That looks like Galacrothe to me. You remember? The Galacrothe that was so
I
common.
actually remember, yeah.
So
Has been a while.
Do you want to look at it?
Yeah, just look at it, not collecting it. It will be enough. Because I actually think that Paco collected that one in the
night.
in the night, during the night.
And there's a sea spider as well.
Sit down to you.
The beautiful red yellow one. Yeah, we came to a spontaneous halt. So now we have a little bit of a sediment stir. Can we stop the movement for now?
Oh, stop.
It's not really washing. Is it?
That's just in front of me. I'm watching this camera, so I'm not moving.
But I mean, it's not really washing the clouds.
It's coming.
I don't know. Yeah.
Have faith.
So I'm too nervous.
It's fine.
Now the view is clearing.
So here have the fish, the
Yeah.
sponge.
When
Okay, I
you-
can start with the.
Do you still need the lasers? Otherwise it would
Let's
be
see how
nice
big is
to
that
zoom
fish.
in on this one.
Yeah, lasers off.
Thanks. It's a nice little community there.
Sure.
Oh yeah.
You hold that for a while?
Hold for a while.
Will you pan slightly to the right and zoom in a little bit?
We're getting some instructions also from our visual and communication colleagues who can see the screen as well up in the dry lab in our office area. How still everyone is just in this picture, like they're staring for a painting.
That's a very nice image, thank you.
Thank you.
It is a very nice image.
Let's see that. I think over there. Two Pokemons.
Yeah. And
The little polymastia. Yeah.
little cute little polymastia sponge in the left corner, and above is also one. Yeah, so they are all just hanging out together. Oh, okay. Now the fish has enough of posing, or not.
I was just getting comfort.
Are those little parasites, you think?
Maybe. maybe, yes.
Yeah, on its tail. And a shrimp also wants to be in the picture. And little...
Hold the camera still if stuff are moving in normally. So if there's a shrimp passing by, don't try to follow it.
Not following the shrimp, going to the parasites.
That's parasites?
Yeah, I think so.
Eating on the fish. Can you zoom or tilt a little bit so that we can see the head as well? Yeah. What are you thinking? I think we can leave it in peace, yeah. Yeah.
Moving on.
Hold on, let me check the sponge.
We distracted Julio from the sponge. Yeah, I forgot about them. The potential Calucothias, the one that are more brownish, but also fan-shaped around the Rosalide sponge. And what is this one?
That's not sponge.
No, is it the bryozoan? Yeah. So we have a big Geodia sponge, first of all, on which then several other sponges, also carnivorous sponges, grow upon, and yeah, these fan-shaped ones.
Okay, I think that we are good to go.
Cool.
Right?
Yeah.
That's it, we go?
Yeah. We can turn the lasers back off, on. Thanks, and continue.
So all these are the same that we collect. Mysterious sponge.
And some Rosalide in between. And what's this association here?
That's a rock.
Only? But there's
Rock
some...
with the geodia on top.
And some of the worms, Cephalidae, carnivores. And this one is a dead sponge? On the side of it? Or one of the Calucothia?
It's a bit hard to say.
Yeah.
It looks like a Calucothia. Several go on the other list. Okay.
I thought I saw ice, but it was just some dark pebbles. Something staring at me out of the dark.
Black coming after you.
I imagined a fish lying down there, but no. I think those were also the anemones that the small little dark orangey ones that we see sometimes, often in between here.
Maybe the alien
Yeah.
following us.
Oh.
How you call because an UFO is when it's flying, but when
Yeah.
it's under the water, it has a name.
Does it?
USO, no.
Unidentified swimming object?
Yeah, I guess.
The USO. Yeah,
USO.
seems legit. Is Holly a user? No, it's
because it's identified.
It's identified, yeah. Another of these cool fan-shaped sponges.
It happened to me today when I tilt the camera to the left, that it goes like that.
Yes. If you're zoomed in too much, it will.
It will.
Yeah. The idea is, if you want to zoom in on that, centre up in the camera
And then zoom
and
in.
then zoom in. Because the camera...
You see, but now it's not zoom in and it's still doing it. Yeah, it's a little bit different than the other days. Yeah.
Yeah.
Has been happening also before. Feels like there is something physical avoiding it.
Could be.
In the steering of the camera or in the zoom?
This one.
In the steering. Yeah. So Julio, you can
Jellyfish.
also probably see it, has his joysticks there for an... Oh, okay. Oh, it's... Oh, like very delicate bubbles. What do you call it? The soap bubbles.
Soap bubbles.
What you do as a child, or maybe when you're older as well, you have this soapy liquid and then you blow bubbles.
Yeah.
Soap bubbles? Yeah, it looked a little bit like that anyway, and I will look it now.
Can we start movement 340 bearing at 0.1?
Who knows? Asconema? No.
Thank you.
I looked the wrong one.
Roselide, I would go for Roselide
Oh, yeah,
because of
it
the
just-
size
yeah,
and
true.
it feels more like
Yeah.
it.
I agree.
It's a fish over there,
Where?
right in the middle.
Ah, the whitish one. Yeah.
No, to the left. Well,
No?
this one.
Yeah, I mean, yeah.
Yeah, well, yeah.
Oh, I'm getting tired. We started our shift at
You see?
2 in the morning,
Now it's-
so
now it's
and now
out.
here it is almost
Now it's
2
out.
again.
Yeah.
If I go to that, well,
Yeah,
now it
and I
works.
can feel it.
Now it works.
Is that a shrimp behind it?
Mhm.
Yeah. Not a small fishy. Oh, much more. Yeah, or at least another of the E-Clouds coming up. Is that a carnivore sponge?
Yes.
And
That looks
like a spider?
A spider maybe, or shrimp, I'm not sure. Tauche, you see on the rock?
Yeah. Mm-hmm.
Maybe it's not.
But it's like,
The good thing is that I have a sample from the night dive.
Mhm. So we can check up on it.
The night or the yesterday.
Yeah, time is very fluid here. Time and space. Oh, another one of the fish. Let's check this. Do you think we're at this corner now? Yeah, it's now getting more square. Yeah.
So it's a bit more than it's more
Maybe
than a half.
here.
Yeah.
So now comes quite a flat area. Maybe we want to speed up a little bit.
Sure.
Yeah.
Can we speed up a little bit?
Yeah. Just tell me if it's too fast.
Yeah.
I don't think you will because it's very homogeneous. Small one.
Yeah, and also curling up, like the ones that the other ships saw in the night dive.
Those were small, right?
Maybe, yeah. Oh. They really like to hang out here on this one. So it seems to be, seems to be an important area for the fish here.
Can we change the bearing to 330, please?
Many shrimp also hanging out here on the ground.
Thank you.
It's full of fish, right?
Yeah, yeah, more coming up. Oh, there's even 3 of them. The longer you look.
4.
Oh, yeah,
Probably more.
in the distance also. Yeah. I first thought, I think it's an anemone probably on it, and that makes it just look like a hole. Yeah.
If I turn my head, turn the head to port, you can then center your camera up and that'll get rid of the shadows and the eye. Will that be okay?
You will tilt port.
Yeah. If I turn to port,
Yeah.
right, now you square up the camera. Like that.
That's not enough.
Of the fish. Yeah. Where do you see the second one? Oh yeah, that's there in the distance, yeah.
They want us to double check what's on the bio boxes for the next switch to tell them.
Yeah. We have a lot of space in the starboard side. 3 are still free, and I think that's it. And more Lysidendorics would be good, I guess.
Yeah.
We don't have Lysidendorics in the last, and just in the fourth one yet.
Well, but I'm more worried about them if they want some crustacean. So they cannot put it on those because it's already amphipods in here, amphipods in here.
And
maybe this one they can. There's a Lysidendorics.
Reader, can we increase the speed to 0.2, please?
And then here, that's it.
Look, there's
Thank you.
the interesting bunches.
Is that too fast?
Thanks. No, it's good. Thanks.
This is empty, right?
Yeah.
And
That one.
we'll put something here. Correct me if I'm wrong. No? Maybe not.
I think that was in the previous dive. I can check. This big Biobox 3. No, we didn't note anything down for Biobox 3. But I will make it more clear here. Do you want to look further?
Well, just tell them that.
More fish and
That's it.
wasabi.
If you agree with me,
Yeah.
these 3 are empty,
Yeah.
and this is empty, right?
Yeah.
And here it's only the max.
Mm-hmm.
And this is what's in here. Cast 10. Cast 10. But yeah, that makes sense. Paragraph or a rock. No, I think it's rock. Actually, sorry. Biggest sponge there.
No, in the distance there. Oh, and then another eel-tailed visits us.
You see, that guy moves to the left. It goes like
The
camera.
yeah. Mm-hmm.
Reader, can Can we change the bearing to 320, please? Thank you.
These two
The
worms.
crusts. Ah, the worms, yeah.
Now this is the anemone.
And a lot of holes in this one.
Now big rock. Big rock. Rock.
Ah. Is that our little pig? We're now at 1,447 meters.
So that's our big.
Yeah.
It's coming up rather steep now.
Mm-hmm.
Will be very steep.
That should be just very shortly. Yeah, that should already be the top. I think. Can we slow down a bit while we climb the slope? Thanks.
Big carnivorous sponge.
A lot of them.
I say what is that?
Looks funny. The big fan sponge on top now. Is it fallen over?
It's the same one.
Yeah, it's just the direction of it seemed a bit weird. And there's a nice overhang of anemones and lots of carnivore sponges and the tube worms. And of course the sea star is there as well.
Inside.
Oh yes.
Stop for the what?
Yeah.
Remove the lasers, please.
Yeah.
Do you think it's finding food in there or just cozying up in the
Sure.
nice soft sponge?
Yeah, sure.
Or hiding from the squid that we saw before that was hunting fish here. Now we also see the many little pretty anemones around. And the soft coral, little soft coral underneath. Yeah. But this sponge has really built a nice...
Nice house.
Yeah. Nice little cave almost. I just now see also that there are sea stars around on the carnivorous sponges, no?
Yeah.
So are sea stars somehow like they eat sponges, and do they also eat carnivorous sponges, or are they like don't like the sticky texture of them? Is it somehow it doesn't seem to bother them,
I don't know.
or maybe they don't know yet that they shouldn't eat this?
I wouldn't go for the carnivorous sponge. I would go for the fluffy one.
Yeah.
But no, I don't know. Good question.
Maybe the carnivorous sponge is eating the sea star here. That would be a game changer. But no.
Shall we keep moving?
Yes,
or-
let's keep moving and leave them in peace.
Yeah. That's the next one.
Thanks for stopping. We can
Okay.
continue, yeah.
Continue. Yeah.
Hello.
Hello, Paco.
Hello, everyone.
Paco and Annalena arrived. The next shift is taking over.
The big one.
The
Sorry?
2 o'clock in
No,
the
I
afternoon
just
to
arrived.
2 o'clock in the
Big
morning
one?
shift.
This size.
Oh,
It's only happened
Same
once.
species we saw last night?
Probably, yeah. And then the thing that you collect, I think is Galocrothe, the fine shape, tiny
Okay.
one. I think
Okay.
it keeps happening all the time. And
You
those,
mean the first one?
the one that is thin,
Yeah. That was a bit transparent, like.
exactly.
But it looks more massive than the fan shape we collected before. Wow. Okay.
We should be here roughly, so
Okay.
about halfway through.
Okay.
Yeah.
Plenty of amphipods.
I think we're reaching this corner now.
Yeah.
So,
And many fish all
you
around.
have very little for samples. This is a little bit crowded, but you have all these three
Yeah.
and this one. And here the two marks are
Done.
You can put things on it if you want, or if they want. But
What
you
about
just-
the slurs?
So the slurps, Asconema with many amphipods, and there is
You want the
Endorids,
lasers one?
amphipod,
Yes, please. Thanks for thinking of it.
So Endorids and the crust, pieces of one of the big ones that we will take it very nicely. A geodia that came by accident, and here also a geodia that came by accident, and this Endorids. No amphipods
Okay.
Yeah.
that I saw. There may be.
But which one? Is there any slope that needs to be is precious, or where we can put something on? Can we put anything on top of like the 4 or the 5, the 3 or the 5?
Just don't put these Endorids on any, because there is these Endorids.
Yeah.
Oh, there's a
Sure.
nice fan-shaped
Other than
sponge
that,
coming up.
You can put things in any.
Okay.
But
As
the
long
thing
as
is,
you are aware that there are a lot of amphipods in
exactly,
those two.
we don't want to mix amphipods from different stations.
Yeah, but
Other than that, for me, it's just not mixing Lissolendorids with Lissolendorids. And because there are Lissolendorids everywhere,
Yeah.
always one. You don't need. But
Sure.
then here you have plenty of space in the bio boxes. Okay. All this is free and this is free.
Perfect.
Okay.
thanks. Thank you very much.
Very welcome.
All
See you
right.
later. Bye.
We're handing over
We exchange.
to Paco and Annalena. Enjoy the rest of the dive.
Okay.
Bye bye.
Okay.
Okay.
Thank you.
Thank you.
Yeah.
See you.
my friends. All the best. See you later.
Hello everyone, my name is Paco. I'm curator at the Museum of Evolution at Uppsala University in Uppsala in Sweden. And
I'm Anna, and I'm from Hamburg, from the Senckenberg Institute. It's our pleasure to go to the next dive.
yes,
Fly along
yes,
at our unnamed seamount.
we were there a little bit last night, then we had a good night's sleep, and we're taking over this second dive to the unnamed seamount at 76 degrees north on the eastern side of the mid-Atlantic ridge, the Arctic mid-Atlantic ridge. We've already got some samples in, but we still have quite a long way to go for this transect. So another couple of hours with us, and some more sampling to explore this never explored before seamount. So we are the first ones, the first scientists and the first humans actually to see a little bit what type of animals and habitats. And so far we've seen already quite a lot.
Yeah. But the real beauty is really shown in the lab afterwards, when we look through all these samples under the microscope and sorting the sediment. There are lots of critters living in there.
Yeah, because we had a first dive already, so some of those samples were already processed and they look promising. This is the second dive we're doing here, and then we might have a third one, so that at least we have sampled several places. We will see.
The samples are so super precious to us because they're on the eastern side from the Atlantic Ridge.
Yes, yes. We know far less about the eastern side of the mid-Atlantic Ridge than the western side. Maybe even we know nothing about the eastern side. I don't dare to say it, but it's a little bit like that.
At least there's nothing published in obvious sources.
Mm-hmm.
So we were unable to find anything.
Yeah, yeah.
That's 005.
We seem to be on the kind of a ridge.
Yeah, it's going down, Paco.
Okay.
Can we start the movement of the ship bearing 000 north at 0.1, please?
Yeah, 000 at 0.1.
Yeah, if not, we want to have it come to the northwest.
So we don't like it when it goes down because it's not easy for the ROV pilots to swim, to fly,
and we see less.
And we see less things. Yeah,
So
it's not good for scientists as well because the way the camera is positioned, it's difficult to know.
Well, and there's a slight upwelling, so we have a lot less visibility for the water.
Yeah. Yeah, a lot of action in the water, a lot of crustaceans.
Yep. And some sponges.
And some sponges at the bottom. Not a whole lot. Some pilolavas. We hadn't seen pilolavas for a while. So pilolavas are those round rocks you see at the bottom, and this is when magma comes up at the surface, meets water, and the magma solidifies very rapidly. So the minerals in the magma don't have really time to grow. This makes a very fine-grained rock that we call basalt. So whenever you find pilolavas, you know this is really the bottom of the oceanic crust. This is the top surface of the oceanic crust. And when you find it on land, it's an indication that there was a sea before here.
Where do you find it, for example, on land?
So on land, we find it in a lot of actually mountains,
Can we increase the speed to
Like the
0.2,
Alps?
please?
like the Alps, for instance.
Thank you.
Change of tape in 10 minutes. I used to be a high school teacher back in the days, and I would take my students to the French Alps, and especially there's a place called Le Chenaillet. We would walk to the top of that place, and we would find pilolavas exactly as you see them here, except they were on land, they were in the Alps, and this is just because the Alps is actually a confrontation of two plates. Before there was an ocean in between, and it was crushed and taken up.
And here the ocean plates are spreading apart.
Exactly. Here it's
So very,
the opposite
very
movement.
slowly.
Yeah. We are on a mountain that is right next, actually, to the Mid-Atlantic Ridge. So this is probably the youngest mountain that was created on this part, on the Eurasian tectonic plate. And as we get far and farther away from the Mid-Atlantic Ridge, from the middle, we get mountains that were created, that are older, that were created a longer time ago.
So one of the questions that we're looking into is if this mountain ridge acts as a barrier to see how things from the east and western side are related.
Exactly.
For this we take samples and fix them appropriately and look at the DNA from all kinds of specimens.
Yes.
Some of them have larvae and some of them don't. So we're looking into how this influences their distributional patterns.
Exactly. One of the long-standing questions in marine biogeography is are mid-Atlantic ridges natural barriers for populations, for species? But for that, to answer that, you need to be able to sample on either side of that natural barrier of the Mid-Atlantic Ridge and
fix them straight away appropriately.
Yes, to try to get genetic data, and we're the first ones to actually be able to sample on the eastern side. So this feels like a privilege to us.
Yeah, it is a privilege.
And this afternoon, Rob is our ROV pilot. Andrea is our co-pilot today. And Patty behind is our advisor, supervisor. You know, I was with a school this morning talking with little kids, 8 years old. I had so many questions on the ROV. What is it made of? How can it withstand the pressure? How comes there's electricity in the ROV if it's in the water? And I wish Patty would have been there for some of these questions. I was like,
be able to answer some of those. Yeah, good questions.
Yeah.
If you have them, Paco, let me know and I can give you the answers for if there's any more classes.
Yeah, absolutely. Thank you. Yes.
Several of the fish.
Yeah. So that's something we noticed actually during our first dive here last night. There were quite a lot of fish on the seamount.
Mm-hmm. Contrasting with the seamounts that we'd seen before, where we mainly saw eggs from skates.
From skates. Here we saw one skate only.
Yeah.
We will need to check the footage during the night, but it doesn't seem like there's a lot of skates or even eggs. We haven't seen eggs at all.
Would it be possible to stop, please?
Yes.
So we are at 1,455 meters depth, and we're gonna take a little stop. It would be an opportunity to study the ground, but also to change the tape in 5 minutes, because we are recording this, of course, and our tapes are 2 hours long. We need to regularly change the tape, and we make multiple copies of these tapes in case something goes wrong. It's always better to have copies of everything in science.
Tape stopped?
Oh, sorry. No, yeah.
Okay.
No, you stop.
What stopped?
What are these potato-shaped things?
Sponges.
Yeah. I'm just laughing because sponges are everywhere. Even if you only see rocks, then high chances you'll see some sponges as well. Anemones.
Do you think we could slurp it? Because we've been seeing this everywhere. I think I know the species, but for this I need confirmation from. I need to connect what I see with the sponge as we get on board.
Yeah, if it's not too far away, yeah, we can scooch forward and get it, yeah.
Yeah?
Which one's that?
No, but I think they're also in the front.
Or some are closer. Yeah, maybe if
Which one?
if you zoom on those, maybe they're the same. I'm not
Yeah,
sure.
That's what. Look at these. Let's look here.
At least they would be easier than to get than these ones, which.
Let's look at this one first.
But
I don't think so. I think any of those, yeah?
yes, but this will be easier to get than this.
Yes, but isn't it easier to take in a shovel?
This will be easier to get in the
Because
shovel.
you can get the little rock what it's sitting on.
Exactly. Or are you thinking maybe the slurp would be easier for these little rocks?
No rocks for the slurping.
No, not for the shovel or slurp.
How big is that rock?
These little
look at
pebbles.
These ones.
Oh yeah, we get those.
Or this one. This one. This one.
Yeah, we get that one on the right there with the scoop.
Yeah? Okay.
Yeah.
Oh,
You want to do that too, or I do?
But can we do a zoom on one of them to have a really good...
For sure. Which one is easiest for you to get?
The one
Like
to the right,
this one?
yeah, that one.
This
Yeah.
one? Okay.
Okay.
Yeah.
Now lasers off.
Yes, I'm gonna wait for...
The rough is moving. So
Yeah,
we
I'm
can't
gonna
do
wait
it.
for the rough to settle firmly on the ground. And anyway, I think we should maybe wait for the tape change.
Okay.
Like this will be no stress. And like this we can do our little zoom session. Is the RAV on the sitting?
Yeah.
Yes. Can we maybe zoom on this? Yeah, on this one. We're gonna zoom. And then there will be a tape change in 2 minutes.
Which tank?
With a shovel.
It's in the bio box.
So if they're picking it up with a shovel,
Don't you want to go on this one so we have that one clean?
3?
Yeah.
Yeah. So this is starboard, right? And this is, I never know the name of it.
I'm also confused.
On the left, what do you call the boxes?
Bio boxes.
Yes, but
Outer.
outer board or
Outer.
outer?
Yeah.
Okay. When we take the shovel, can we try and get these crawling units in. Just wait for a second, it will walk towards the sponge.
Will they not fall through the holes and scoop?
Well, we could try.
Yeah.
Tape change in 1 minute. So whenever you're ready, Andrea, we can do it now.
1, 2, 3, stop.
Stop recording.
Well, this crawling unit there is an amphipod. Yes. You ready?
As soon as the new tape is on.
So while this one amphipod has crawled out of the picture, the one I'm actually looking at is one walking towards the sponge.
Recording.
And now behind, no, look, now it's going directly to the sponge that you want to sample.
Oh.
So.
Perfect.
Well. Okay.
Okay. So this will be cost. Yeah, I'm good for the image. Oh.
Okay.
But maybe we can see it.
Well, that's tricky now to see on this camera what we're doing.
How is it, Rob? Can you actually see what you will be doing with the scoop?
Yeah. Move the drawer back in.
Okay.
Okay.
Yeah.
So I'm writing the date, coordinates.
Can we
Is
stop
it a
the
zoom in the
movement
HD?
for now, please?
Keep going.
Thank you.
We are going to sample the little sponge sitting on the pebble, because it's a little bit everywhere. It seems to be quite a common species, but one of the main problems we have with sponges is that they never look the same when we bring them on deck because of pressure, because they're out of the water and so on. So we have a lot of difficulty connecting the species that we collect with what we see on water. And here we're privileged enough to have ROV, high quality, high resolution images that we can actually connect to the sample. So it's fantastic for us to be able to say, okay, this thing, we know what it is. We're collecting with a scoop. The scoop method is very practical.
Where am I on it?
Yes, you're perfectly in front of it.
Yeah, where is it?
Can you show, yeah.
This one, the little white one. Just if you shove it from a big bag, we'll get some rocks with it. That's fine. There are a lot of little encrusting things on the rock, like small anemones and some polypid worms, so we don't mind when you get a few rocks.
Yeah.
Of course. I will write plus pebbles.
Okay, zoom out.
I'm zoomed out, Max.
Fourth drawer.
Outer board, yeah. Outer side.
Upper side.
And bottom left. Here.
This one.
There's nothing inside. We cast 14. it's good to see. The mug hasn't gone there. That's interesting. It means it can stay actually if it's clean.
Has it been used? Both mugs are full. Yep.
Yeah, this one has been used, but not this one. Here, there's nothing. But the mugs have been used. Yeah, they're full.
Yes, but if you want another sponge piece, you can put it on
I
top.
think so because it looks clean.
So if you have a picked one,
Yeah.
that can go on.
Okay, cast 14 is in. Yep.
Is that everything for here?
Sorry?
Is that everything for sampling here?
I'd love to have one. Is it enough for sampling? Yes. I'd love to have one look.
Yeah, we
Just
can
to the
have one
front.
more look, especially if you see something,
Yeah,
on
but
other pebbles or.
Yes, but I want this to be stored first. Zooming in, out, anything?
No, I just want to know what you want to do, that's all. Do you want to zoom, do you?
Oh, I want to zoom,
Yeah,
just to look. Just leave it out in case. Discover treasures. Sorry, my eye is so focused on amphipods, I only see them everywhere. Or the only thing that's moving on here. Nee, fly on.
I mean, I would be interested also to get this little sponge. If you
Can
think
put
there,
it in the same box?
we can absolutely
now
put
that
it.
the shovel is out?
Absolutely.
Okay.
Can you zoom on it
I'll try.
fully? But maybe you're already at maximum.
I'll try to get it set up. Just this is.
Yeah.
Sorry, we'll go back to the sponge in a second.
Sure,
I'd
sure.
just like to have a look here.
Do you think it actually sensed that we were opening maybe a place to find food or?
I do not know.
So I hope you can all see on the left this little amphipod, this little crustacean walking on the sand, on the mud.
Well, excuse me.
You can see the bright eye, or I guess they have two eyes.
They have two eyes, but in some group, this is molded together.
Oh.
It's like a cyclope.
Mm.
So it appears to be one eye, but it's actually two.
Very nice. We can do a little bit of behavior observation. It's very rare that we can see those animals in the wild behaving.
I'm still impressed by that, being like 1.5 kilometers below us, that we can look at these little creatures
Yeah.
sitting on this vessel and looking at something that's 2 centimeters in size.
Absolutely. The little, so of course I'm looking at the sponges next to your amphipod, but the little white sponges I think are actually Tentorium. But I think we'll try to collect them as well for confirmation. They're a rather common deep sea sponge in the area, all over the Norwegian and the Greenland Sea.
Okay. A few seconds more, and we will take a shovel again, and we will go into the same box as we just did.
Right.
But we will take this white sponge that Paco will show you.
Yes.
This
So
is
I think it was, yes, one of these white ones. So it's next. You have the this, and it's right next to it.
exactly. You
Yeah.
follow it. That one.
So just leave the lasers there on it, and we'll come down to it there now. Just leave it there as it is. You come down. So it's just to the top of the laser, Andrea.
Okay.
Yeah, exactly. It's very close to the laser. So we're still using the scoop. The scoop is a really practical tool, especially when it's kind of soft and we don't have to break into a big rock. And then at the same time we can take some sediments which will be sorted on board later on. Nothing will be lost. Everything will be studied in the lab and processed.
It's going into it, is it?
No, it's a lot.
Right?
It's in.
Yeah.
Yeah, it's in.
It's in. You can even move a bit back.
Yeah. Don't take too much because we don't want to
We don't.
Yeah.
cover everything in the bio box. And
It's a little problem now.
yeah, I don't know if you can.
What's that, Andrea?
Can we move back a little bit with the shovel?
Thank you for.
Is that
Or can you shake a little bit the scoop
Yeah.
to get rid of some of it?
Yeah, which means instead of it blowing,
Towards the other way.
let's go. Very sorry.
Look at this output now. In between its two places, we suddenly
Oh yeah.
things changed.
Oh yeah. I
I
see.
see the shaking.
Yeah. So we're shaking a little bit the scoop to get rid of a couple of
Mud.
stones, some mud. Again, to give you an idea of size, the lasers you see are 10 centimeters apart. 20.
Well, you have conflicting information.
Mm-hmm.
Can we ask the master? 10 or 20? 10. Okay. Sorry.
No,
we were told 20. That's why we kept saying 20. This is yesterday. We got a 10. That makes a lot more sense.
I'm going to turn it towards the camera, and I think it went in the back left, either right hand side.
Fall.
Still too much sediment in our pack, though, is that?
You think? I mean, the risk now is that we lose the. I think the sponge is inside. I think.
I think it went back in the left corner.
Yeah, exactly.
Mhm.
So it should be okay.
Do you put this much into the buy box?
It's not
What do you
this
think?
one.
Yeah.
Oh,
Sorry.
just a second, Johnny. I'm not entirely sure. I thought it went in.
I think it is in.
And it's at the very end of the shovel.
Yeah.
And I think, yeah, just
Oh, good.
we lost some mud, which is good. The same as before.
Yes.
Port roll on the back.
Are you taking more sediment?
No.
No. And then we
Our
have
part-
enough.
I think that's fine. It's
Okay.
Because the other parts in it are the coffee mugs, and they're separated by the
Yeah.
mat.
Yeah, but then
net.
we shouldn't put more there. I think that's it for that bio box, right?
Otherwise,
It's because it's going to be quite muddy.
this will be
Yeah,
a
I
lot
can give
of
it a
mud.
shake, but not out. Like I'll let the sieve of the holes do the
Mhm.
sieving.
Yeah.
So if you just
Yeah.
come left with me a little bit. get out and so it doesn't totally obscure our vision.
You're doing a great job.
Yeah.
Jenny, this saves us hours.
And clean windows as well.
Well,
So we're trying to get rid of the really fine sediment. This is something we usually do on deck. You usually do on deck with the sieves.
it's necessary to get the small specimens in there, but we have a lot of samples where the small specimens are actually kept in the net, and these are attached to a coffee mug, so-called, where you can see the handle, the bottom left. Und dann tauschen wir dann immer so. Oder wenn einer von uns eine Roboter probiert, das ist das andere. Ja. Okay.
So we're good at that, Paco, is that?
Yeah, that's good enough. And that is, yeah, the bottom left. As you can see in this drawer, we have 2 mugs. They've already been filled. We have a sponge on the right, and we're putting now our scoop contents on the left, bottom left compartment. Perfect. Splendid. Thank you.
Thanks a lot.
I think that's it. A little look inside. Okay. Andrea?
For me, it looks empty. Do you want to
Yeah.
check?
Can you
If
take
this
them around?
one's-
Looking good?
It's looking good. Yeah, completely empty.
Yeah. Okay. Let's throw that away, so.
Yeah, you can store everything. Thanks a lot.
Okay,
Yeah.
standing.
So after this is stored, we can fly on.
Yes.
Leaving confused afterwards. We often look to our phones because we get certain sampling requests
Yes.
from colleagues all over the world.
Or comments on what we're looking at, and
But sorry, we can't sample that much.
but I mean the layer of pebbles is really thin. It's just one layer of pebbles, then it's mud. And I don't think we've ever seen this type of ground yesterday.
No, we haven't.
This is a bit new.
Zero, more or less.
I'm missing Bodel sauce.
Can we start?
Yes, please.
Yes.
Start, please.
Start on on, is it? Okay. Bridge RV, can you resume the move there, please?
Doing move 000 at 0.1.
Yeah.
Our pilot is now Andrea and co-pilot Patty.
Fish.
Temperature in the water is minus 0.4. It's a little bit colder than yesterday. Oh. I don't know what that is. If it's a lot of sponges or worms.
We're looking at this rock with crossing units towards us.
It looks like worms. It seems that every time you have a little surface, the worms really stick to it, but always on the same side.
On the same side, yeah.
Yeah.
Exactly, on the one side.
Like here.
Yeah.
There's a hairy sponge coming up. That's probably a geodia. They are very abundant on the cliffs around. Yeah, there's a couple of geodias here. And the little soft coral. And then the orange colors you see are little anemones. The pink ones as well.
And a boat, like a snail sitting on there.
Oh yes, absolutely.
So, Pepper, the next one, shall we just stop in front of it and have a look?
Mm-hmm.
Might be a bit late now.
Okay, Bridge RV, we started point what?
Point 1.
Sometimes we see this dark brown patches in sediment. From looking at samples this morning, there was a lot of rhizomes, but this doesn't quite look like this. Do you mind stopping here so that we can have a closer look? Thanks. Thank you.
They do seem like, because this is the bryozoan, I think, and then here are dead bryozoans.
So the dead rhizomes will be the brown ones, and then you have the white little ones living on top. So rhizomes are lace animals.
Yeah, they are. Yes. Also filtering the water and eating in the water column. It's a large group. They're usually very small animals, but they're very important. They're everywhere.
And there are very few specialists.
Mm.
Well, there's a very nice sponge, the white one.
It's a,
This little amphibot just ran behind it. I was hoping it would come to the front. It just disappeared. So, an enemy.
the little round sponges look like golf ball sponges, but they're not. They're young geodias growing, which means that they like actually to settle on this mat of, maybe it's a mix of spicules and bryozoans and so on.
Okay. Shrimp. We can continue. Thank you. Yes. Have you looked to see stuff?
Nope.
RV bridge, let's resume the move, please.
So constantly we have to talk to the bridge on the boat so that the boat follows the
No problem,
ROV.
thank you.
We are connected through a tight cable, a very strong cable, and the ROV and the boat move hand in hand. When the ROV stops, the boat needs to stop, and so on. And we are in the command center. We call it the ROV shack, where the pilots have a number of screens and cameras showing them a little bit all around the ROV. And we, the scientists, can see the main camera feed. And we can control that camera to the right and left, and we can zoom in or out to make our observations.
But when we zoom in while flying, we get dizzy. Have to avoid that.
But we're very lucky with the sea conditions, I have to say.
Yeah.
Outside it's pretty flat.
Okay.
Almost no swell. Meaning
No. No, let's fly on.
if the sea conditions are too bad, we cannot dive with the ROV. So for us, it is essential to have good weather conditions.
Can we stop in front of this rock, please?
Okay, bridge RV, all stop.
All stop.
It's quite incredible when you think of it that we are making a 65-meter long ship stop because we want to look and zoom in on little worms at 1,400 meters depth. That's quite a lot of logistics and equipment, which makes those observations quite precious.
Now. Now, what do we see?
Yes, I see amphipods, of course. I see.
I see sponges.
Yes. I think I see two amphipods.
Oh, there's one moving right
Oh,
across
yes.
the screen, just
Yes.
so you don't, it's not forgotten.
No.
And you see one sitting at the bottom of the rock
Yep.
on the left. And you see one like 10 centimeters on the right of the rock. You can, when they're alive from this group, they're often reddish and a shiny eye, so they can be spotted easy.
Mm.
These
Oh.
are relatively large ones. There'll be plenty more smaller ones.
There's a question I wanted to ask you actually. So I remember in the previous live you said there were 10,000 species of amphipods, but how many species in this area?
What is happening? What is happening with the light? We're not doing anything either.
It's flickering a little bit.
Shall we fly on?
Let's wait.
Yeah, no, but
for them.
I think it's worse when we zoomed in.
Yes. Should we move on?
Yeah. You can turn the lasers back on, please.
I try to log really every fish we see because fish are one of the few groups where that can be identified on video. So by analyzing this video, fish experts will be able to tell something about the populations of fish on this unnamed seamount. We cannot do this very thoroughly yet for sponges because many of them that we see on the ground cannot be named. That's why we're trying to subsample some of them at least to try to get names so that people can analyze those recordings much better in the future. This fish is not afraid.
Bridge RV, can we resume the move, please?
000 and 0.9.
Thank you.
There's a little carnivorous sponge at the bottom, this little bush-like structure. But we've seen places where they were truly abundant. On the cliffs of this unnamed seamount. That's a fish, okay. Wow. So we're meeting another cliff here. On this cliff we have two species of geodia, Geodia parva and Geodia hancheli. These are typical Arctic species.
Now let's focus on the sea spider. Just here. Can we stop here? Yeah? It's just because it's right in the middle of the screen now. And they're so colorful.
Stop.
Give it a look.
So people have been asking on social media how large these sea spiders are.
You see the laser now, 10 centimeters. So what's it, 25? So the distance between the two pink dots is 10 centimeters.
Yeah.
And you can see the sea spider. The legs are
more
larger.
than 10 centimeters, the legs.
Exactly. So it's around
maybe 15? And
yeah, 15 centimeters a leg. Your guess is as good as ours, yeah? And so across it's 30.
That's what I would say.
Yeah, it's quite large.
Which is very large indeed.
It's our lower arm.
Yeah.
It's from. And they're very pretty creatures, I think.
So, could you tell us, are these crustaceans or no?
No, they're not, but they're related to spiders. But when we have meetings talking about news, scientific news, then the sea spider people come to the crustacean meetings because they're more crustacean people studying the marine environment. So to learn more about interactions and influences from habitats and distribution, there's more similar methods to the people studying crustaceans.
So are these actually spiders that went to live in the ocean? Are they like arachnids? Are they part of the
could it be the other way around? Could it be that, well, I think that all life evolved in the ocean, but I have no idea if these were...
If they
first.
came back or, were originally in the ocean. Okay, wow. And is that a typical Arctic species, the one we're looking at? Yes.
It is. They also go to the size in the Antarctic, but there are quite a few around which are much smaller and not as colorful, but not less interesting. So we'll do just go down a little bit and zoom in here.
We can see actually that the worms are growing really on the pillow lavas, basically. And that's where the geodias and the anemones are also.
And the ascidians.
And the ascidians,
there are quite a few ascidians here. Ascidians are this translucent objects with the two openings. Look a bit like a balloon with a hole.
Yes.
Still inflated, a lengthy.
And all these organisms feed in the water column. They're attached to the bottom floor and they feed on whatever the current brings to them. For the sponges, it's really small things, really small living organisms.
Look, this anemone just ate an amphipod.
Really?
How dare it? Here on our on the bottom right.
Yeah?
Yeah.
How do you know? Did you just see the amphipod?
No, I'm guessing because I just saw something light in there, so I thought it's the eye, but
Mhm.
I might be mistaken. And I have seen them doing, I mean, that's what they also feed on.
Hmm.
This is of the lower ones, just had a meal, and they're putting in their tentacles. That's how they feed. Their mouth is right in the middle. We're not actually moving anything with our camera, it's rough moving, which is good.
But you can tell there's a lot of current. I mean,
Yes.
this is not current created by us.
No.
This is natural current on this wall.
I just wanted to look at the 6 months again.
It's not moving that much, right?
No.
Is it
No,
because it's waiting for a prey, or?
Come on. You're asking
No,
me
but
the
I would expect,
psychology of
I don't
6
know,
months.
that they're crawling on the floor
Well, they sometimes
with a
do,
mission.
but give them a break. You know, just waiting for something to come along.
Mm.
Yes, thank you. We can fly on.
But look, there's the pillow lavas, then there's
You can still fly on.
the tube worms, and then there's a crust of
Yes.
mud.
Yes.
It's like 3 layers.
Yes, but the anemone made it to
Yes,
hold on to
the bedrock,
yeah, or yeah, maybe
or
there's a little
even
bit
the
of-
mud, actually.
Yeah, but it looks like holding on to the mud, but
Yeah.
it can't be. That's very nice.
It's very nice.
Some
Oh,
fish.
there's a fish here hiding. A lot of carnivorous sponges as well here. All the bushy white animals you see are carnivorous sponges, so they are not filter feeding in the water column. Instead, they are probably catching some amphipods or other crustaceans.
They will be. That's why these amphipods are so important, because they play such a central role of eating and being eaten.
This crust is going a little bit started now.
Pretty sure everything else up.
All stop.
So I'm going to repeat a little bit for incoming viewers. We are on an unnamed seamount that is parallel to the mid-Atlantic ridge, but we're on the eastern side of the ridge, and this is a huge mountain. We're talking 30 kilometers long mountain, unexplored to this day, and we're the first ones to see the animals living here. There's a ctenophore, let me type that. The summit of that mountain is not very shallow. It's at about 1,200 meters. So we're actually maybe 200 meters from the top.
Yes. We're 1,437 now.
Yeah. And one of the questions we're trying to answer is, are the communities on the eastern side of the Mid-Atlantic Ridge comparable to the communities on the western side that we've been exploring so far? And I mean, it's difficult to say right now, but there are a lot of common species. The geodias are one of those, the white glass sponges. But we've seen a couple of new players here, like the small soft coral, the worm-like mollusks called solenogasters. So there might be some differences. Analysis will need to be done. Samples will need to be processed. Specimens will need to be identified. And we will tell you, but it will probably take weeks and months of analysis to be able to give a straight answer. And most of all, we will need to do genetics, because we want to see, we want to know if the populations we're catching here, subsampling, are connected genetically to the populations on the other side of the mid-Atlantic ridge.
I like all those fishes sitting in between.
Yeah.
And there's a shell, well, like a garden snail, related.
The lasers are 10 centimeters apart. So this fish is about 15 centimeters long. And you can see the geodias, those furry balls, are for most of them 10 centimeters, but some of them are definitely 20, 30, 40 centimeters large. And there was one paper 2 or 3 years ago in Nature Communication that tried to date the geodias, saying that a 20 centimeter specimen was about 200 to 150 years old. So these animals probably grow very slowly in the Arctic, and most of them are probably very old. So we can't just speculate on the age of a specimen that's 40 centimeters or 50. So these are maybe 500 years old.
Right, Jarvie, can we resume the move, please?
This old age is also true for some of the hard corals we encountered.
Mm-hmm. Yes, that was during our bamboo coral dive.
Yes.
A few days back.
And there are different methods of age dating, aging specimens. Some of the corals, they have like rings, like a tree.
Oh.
You can really count.
Mm-hmm.
Yeah, count to the hundreds. So you know they're hundreds of years old. That of course only works when there's like a shift in seasons.
Just like in trees.
Yeah.
Seasons,
but for many of these invertebrates that we see here, we have no idea. Many of these actinaria, these anemones, we don't know how old they are or can be.
And amphipods, do we know how long they can? Are they annual organisms or?
No, but we don't know for the deep sea ones. There are also quite a few from lakes. It's like glowing, look. And those can be held for years in the aquarium.
Okay.
And in the deep sea, especially in the Antarctic, they can grow very large. And they have to molt. So they get out of their, their cuticulars, like skin, that's limiting their growth. And then they're very vulnerable directly after hatching. Hatching the wrong term, but molting, get out after the molt.
So we don't know, but probably several years.
Yes, many years. Definitely.
Have people been able to raise them in aquarium?
Yes.
Wow, like reproducing.
amphipods, yes.
Yes?
Yes, yeah, for sure.
Deep sea ones? Or only
There
shallow?
are attempts from deep sea ones, but when they get too stressed when sampling, they sometimes release their juveniles. So they haven't come into the next cycle of breeding. But the shallow water ones, that has been done quite successful.
Okay.
They're also used as fish food. So there's a commercial interest in these.
So I have to say, for deep sea sponges, it's very, very complicated. I don't know of any aquarium like open to the public that has really deep sea sponges to show, or they're not very, very deep sponges.
Most of the amphipods are too small to be shown in aquaria to raise public interest.
You need to get the really
yes,
big ones from the abyss. Yeah.
They don't have a swim bladder, so they don't mind too much getting up. Well, and that's also the wrong term, but they survive. Our difficulty here is to keep them cold. So we keep them in cold seawater. We have a photographer on board who is able to take fantastic pictures. And the specimens still have their live colors.
What's that?
And have all the appendages intact.
Yes.
His name is Solvin.
Yes.
He's doing a great job. So we're trying to avoid like getting air bubbles into a sponge, for example, on board.
Yes.
Keeping the animals cold is essential.
Yes. So when the ROV comes back on deck, we usually prepare buckets of very cold water to welcome these animals and try to keep them alive a little bit longer, so that pictures can be taken, observations can be made while they're still alive. We also want to preserve the DNA as long as possible, especially since we're collecting also for RNA. RNA is a molecule we all have, but it's a bit more fragile than DNA, and you have to sample it ideally when the animal is still fresh. Exactly. So when I put a sponge in alcohol, it will extract a wide array of compounds. So I keep that ethanol, I call it the first juice, the first extract. I keep that first extract preciously because it can be studied for all the molecules it has extracted. We can look at the different masses of these compounds and maybe find something new.
Speaking of ethanol, this is a dry ship. And the ethanol that we've got, well, it's poisoned. It's wrong.
Oh yes.
So chemicals, compounds are put to it that it's good for preserving, but you wouldn't be able to have any yourself.
To drink it. Yes.
Are the lasers on?
Yes.
They are.
So
downhill. It's hard to see on this one.
So we're in the blue, as we say, when we fly over a little bit higher because the terrain is either going down or is it a little bit complicated to handle, then we go in the blue and fly over to another place that's more suitable.
So overall we're following a transect, because while this area has not been studied before for any biological content, it has been mapped to a resolution of 35 meters. Approximately. Yes. So we have some unforeseen dips here.
And we were talking about chemicals extracted from sponges. So there's actually several drugs on the market that are either inspired or that have been extracted and discovered from sponges. Two drugs are currently on the market called vidarabine and cytarabine, used as anti-cancer, against leukemia, lymphoma, or also against herpes viruses. And these drugs were extracted from a sponge from Florida, from the tropics, in shallow waters. So this is just to give you an idea of the importance of doing chemistry analysis on sponges. They can lead to really interesting drugs for us.
Yeah, not only drugs, they're also used like in the anti-fouling industry.
Yes.
So there's a lot of research going on.
Yes.
What can be extracted as a natural compound.
Yes, exactly.
Like to put on boats so that other things don't settle on the boat.
Yes, it's an opportunity to find natural compounds from nature that we could integrate on boats to prevent fouling. What we call fouling is when other organisms grow on the hull of the boat. It's a huge problem in boat industry. A lot of money is lost because you have to clean regularly the boats, the hull of the boats, because when things grow on the hull of the boats, it slows them down and you lose money, of course. If your boat
you require more petrol too.
You need more gasoline and so on. So there's actually a lot of compounds extracted from acidians or sponges that are currently tested in boat paints.
And sea cucumbers.
And sea cucumbers. I mean, all these animals also don't want anything growing on them, so they have already developed through evolution.
Some of these sponges don't mind, but some are very clear.
Yes, yes. So yes, there's a lot of biotechnological applications for chemistry extracted from marine organisms in general. We call it the blue economy. The blue economy is everything that basically people can make money out of the ocean, and this includes all this blue biotechnology field.
Well, sponges are used commercially as bath sponges.
Yes, that's probably their oldest application.
Yes, the ones without any spicules.
Yes, since the Greek times, sponges have been used, and that's why there's a word in Greek for sponges, in Latin as well, spongia.
Increase point 2, please.
The Latin word for sponges is porifera. That refers to all the pores they consist of.
Yes. And the pores are there because they need to filter water, and so they need for the water to come inside and also to push it outside once they've eaten what was in the water.
It's a bit strange how they eat. I mean, they don't have any organs.
Yeah.
They just have pores.
They just have pores. There's no organs.
Different kind
That's
of
true.
cells.
So no digestive system at all. Just canals bringing the water to special little chambers, and in those chambers they have special cells that will pick up the food, not just any food randomly, but they really select what they want to eat, and then they will just let go everything that they do not want, and yeah, it works
through one opening.
very well. Sorry?
They let go through one opening.
Yes, then the water gushes out from what we call the oscula. That's the main opening through which the water comes out of the sponge. There can be several of these main openings, but usually when you see one central hole in the sponge, that's the oscula.
around forever.
And they've been around, so not as much as
Not
bacteria.
as forever, sorry.
Not as much as bacteria, but
dinosaur level.
yeah, way before the dinosaurs. So the dinosaurs appear in the Trias, the first dinosaurs, and sponges appear about, I would say, 300, 400 million years before that. So they are, they emerge way before the dinosaurs. But they were also living at the dinosaurs' time, the Geodias. So for instance, so one of my studies, I looked at Cretaceous fossils of Geodia. So they were there during the Cretaceous and the Jurassic, probably. So Geodias were already present in the seas, side by side with dinosaurs.
So they're fossils that were found, or how do you know?
So we have two types of fossils for sponges. We have fossils from the spicules, which is the skeleton, the glass skeleton of sponges. And we also sometimes, more rarely, we have actually whole sponges that were fossilized. But it's a peculiar sponges. It's what we call the rock sponges. They're very hard, and therefore they fossilize much better. There's a nice fish. So the fossil record for the rock sponges is very good. And these rock sponges actually start to appear also during the Jurassic. We have actually sponge reefs of rock sponges at that time. They're very important in the Jurassic underwater world. Not necessarily. I think the white spots are probably the one we collected earlier. Maybe some are snails as well, because I remember that we also looked at some in closer view earlier. There's a couple of carnivore sponges, like one right now, a little bit to the left of the screen. But it's essentially paleolavas with a bit of mud.
Covered in mud.
Yeah. And are we going down? Yeah. So we have a bit of a passage going down now.
Finished.
Okay. Let's try to plan a transect going up, because we know it's much better for viewing and stopping and exploring in general for the ROV. But for this unnamed seamount, we have maps with a resolution of 35 meters, meaning that every 35 meters we have pixels basically that are the size of 35 meters. And although that may seem quite precise, for other maps we have 1 meter resolution. So of course we know far less, and it's much more difficult to plan a transect here in this unnamed seamount because we have this uncertainty.
I would actually like to stop and look at this.
Next to the sponges?
Yes.
You might see us regularly looking at Andrea's screen, because she has a screen that shows us the below the ROV, which is really useful because it's good for us to know where she can land with the robot.
Or just hovering. You don't have to land. It's more stable landing, yeah. See the little shrimp here on the right in pink.
lot of crustaceans in the water. It's very rich.
And also several shrimp sitting here. See the pink ones?
Mm-hmm.
And little amphipods. And what are these? Actinaria? Sorry, I'm zoomed out.
Are these amphipods sitting on that sponge? Yeah.
I would guess so, but I'm... Yeah. I can't see close enough to confirm, but yes. I counted 11.
There's a little gastropod, but basically I'm seeing the same little sponge species that we've previously collected.
I'm trying to center it, but I can't work anymore.
Oh yes, okay, I see it.
Some anemones feeding posture, and others have their tentacles in. Yeah, we can fly on.
Sure.
Thank you.
Wow, look at that amount of
Stuff.
Yeah.
Sediment.
But a lot of living crustaceans. This is a very rich environment, which should attract a lot of fish and squid and predators. So the water temperature hasn't changed. We're still at minus 0.4 degrees at 1,450 meters depth. I was discussing with the classroom this morning, and I had another interesting question. I had loads of interesting questions, but one of those was, how do the animals get their oxygen? And it is not obvious that when you're very far away in the deep sea that there's oxygen, but there is. It's in the water.
It's essential.
Yeah. Without oxygen, none of these animals could live. But then you use different techniques. I mean, the crustaceans will have gills, like the fish, and for the sponges, they will get it through diffusion. So the water coming in their canals will be water with oxygen, and that oxygen will get into the sponge, and it will be brought to the cells, directly to the cells. But yes, in all of this mass of water, there is a lot of oxygen.
And in this part of the world, it's also a lot of salt.
Yes. Yes.
Great RV.
page.
That's why it's so-called heavy water.
0, please.
It's quite salty. And the different water layers, the more salt, the colder, the denser it is, and the heavier. Therefore it goes.
Exactly. It's not known, maybe very well known to the public, that water can have different weight depending on its physical properties. So depending on the temperature, like you said, and the amount of salt. And cold water is more heavy, and salted water is more heavy, it's more dense. So it's going to go below another mass of water that is going to be warmer and with less salt. And
it's a conveyor system.
Exactly. So
conveyor belt all around.
This creates
the
currents. This creates currents, and
it's also our Gulf Stream. People will know that the Gulf Stream.
The Gulf Stream is well known to the public, and it is warm, less salted water.
So it can only be moved up because the salty heavy water moves down. So like a conveyor belt.
Exactly.
With 1,456 meters.
Yeah. So if you're not familiar with the deep sea, it's not a homogeneous sea. There's actually different layers of water, and it's sometimes difficult for animals to move from one layer of water to another. There's different compartments, so it's not you're not as free as you think you could be when you're small, especially if you're a small animal, and you're dependent on currents and water masses that are sitting on top of each other depending on their physical properties.
Sometimes the horizontal distribution is much wider than the vertical one, so they can be, well, speaking of amphipods here, they can be found 1,000 kilometers apart, and even the same species in the Pacific and in the Atlantic, which is quite impressive.
It is.
That they're brooders, so they don't have larvae, but they are not found in like 500 and 4,000 meter depth. So that they have a vertical limit, for example, between 2,000 and 4,000.
Yeah. So I'm going to say it's exactly the same thing with mice sponges. We have the same phenomenon with widely distributed species if you stay on the same water mass or depth, but as soon as you would go shallower, you'll have different species. So these boundaries are real.
We only look into such selected samples that there are always these questions, how do they get there? But maybe they're everywhere in between, but there are way too few samples to say that.
Yeah, we've explored only a very, very small part of the deep sea. So I've seen numbers like we've explored 10% of the deep sea, which is nothing. It's maybe even less. And if you consider the deep sea is more than half of our planet, well, more than half of our planet has not been explored at all.
I think a little bit of different numbers. Think of the Earth as 3/4 in water, and 90% of that is considered deep sea. So everything below 200 meters is deep sea.
Yeah.
And then it's less than 1% explored of that vast area. So explored to a level that you know what kind of species live there. We're not talking mapped, we're talking biology. That's why it's so likely to find species that haven't yet been described, that are new to science. When we look at the deep sea small stuff, especially.
It's difficult to access, and there's actually few scientists, few specialists for every group.
A so-called taxonomist. Well, it takes time, and it's not taught at universities a lot.
Yes, I would say a lot of genetics and genomics have been kind of replacing the older knowledge, or the zoology and the botany courses that used to be more common before.
But what we're doing here is also creating baseline data. So for further studies, we have images, specimens, and the genetic reference. So when it's found somewhere else, then you can compare the DNA to know if you've got the same specimens.
And it's very important that the specimen, before being sequenced, has been identified by an expert, so that the genetic sequence going on internet has been really verified, so that you can trust it, so that it becomes a reference sequence.
But these open access databases have improved massively over the last years. Last 10 years. The museums, with their reference databases to what's there, as well as the genetic databases, a lot of them are open access.
Yep. And that opens the door to environmental DNA, because environmental DNA is extracting the DNA from the environment. It can be sediment, it can be water, it can be air, it can be an animal, and picking up all those bits and pieces of DNA in there and sequencing them and trying to tie them to a species name, trying to see from which organisms they come. But to be able to do that, you do need a reference database.
So there's no point having a lot of sequences and you know, oh, these are 20 different things, but you don't know what they are.
Absolutely. Yes. So our job as taxonomists is making sure that the reference databases of our organisms are correct. And for that we need to identify and work hard to make sure new species are described as well.
I once was invited to a workshop, and it's the barcode of live databasing.
Yep.
So just to get good reference points, so to look at them taxonomically, and these were for amphipods from Norwegian waters.
Oh.
And while doing that, we found a new species.
Okay.
And we described it, and we named it for Angela Tandberg, who's also on board, expert, and it's quite nice that this species is now found quite often.
Okay.
So once it has a name and a reference, and
then people can identify it and find it. Absolutely. Otherwise, it's invisible.
Yes.
Yes. Naming animals and plants, any living organisms, is essential to make it visible to science, to the public, to policymakers.
It's very barren.
Yeah, and we're still going deeper.
Didn't you tell us it's soon over this downhill path? Almost there, they say. It's a bit like
So we're almost
kids on a long car drive. Almost there. Oh. I see a lot of blue.
So again, two fish next to each other. That's a family.
No.
That's those guys at the end of the line there.
Okay. Now what do we do?
Is it 3 fish?
Yes.
Oh my!
Look there. Look.
Oh. Are there others?
No, I think these are sponges.
Oh, okay. But still, 3 fish next to each other, that's something. It's true.
Can we go up sideways? I'm not seeing this wrong. We're at the end of the transect, yeah?
We're at the end, yeah. We've come down.
So what is the best view
Best view for
looking at this camera? Looking
turn around.
turn around and up. But then we fly over the same area.
Yeah.
We've just
Ready, Jorby, I'll stop there.
Can we sit down
Sure.
here? Yeah. Just turning around.
Do you
Yeah.
mind if I go into the dry lab and try to get some information?
Shall I go? Or do you keep on talking?
I can keep on talking, no worries. Yes.
And do you
So,
have a preference for sampling?
no, not necessarily. Okay, so we've arrived at the end of the transect, and we need to make a decision on where to go next. We're going to consult the cruise leader and other scientists that are looking at the maps as we speak to try to see which part is now a priority for us. And these moments are perfectly normal in exploration because we sometimes rely on what we're looking at in real time to decide where to go next. Could you enlarge a little bit the map?
Larger or squeeze it out?
Squeeze it, yeah. Yeah, thank you. Is it continue? Does it go down, 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 or probably?
It was at 50 a while ago. We were coming along the top here. When we were back here. There's nothing. And as you can see in the shading, it's getting held down. So it's broken anyway.
But this part might be a bit higher.
Maybe there, yeah. But you gotta go down here. Come up. Go down.
Yeah. And how far are we from this part? Okay. Nice. Could this be like a blue, or maybe we have information? Could be. We finished the dive here? Okay.
Shall we do some more sampling? We've still got
I'll bring back a tea when you're coming in. No rush.
But so if we want to do sampling, we would need to turn back, like Patty suggests, because we would need to be uphill, I guess. But I don't know if what we've seen so far.
That has been a bit uninspiring.
Yes, because most of the little sponges we sampled.
We already sampled.
Amphipods we did.
Yes, I would have loved to get some of the Solenogustus that they had, but I didn't see any.
What about push cores? Should we maybe try to get push cores?
No, they've already taken those.
Okay.
Just before.
Mm-hmm. Should we
just turn and see? Just
exactly.
turn and fly a little bit to see if the view is completely different when going uphill, and then we'll finish the dive.
Yes.
Is that okay, Paco? Paddy?
To go back over the line we came.
Yes.
Yeah, or
a little bit.
Yeah, or
and see if we can sample something else.
The other transect is too far away. It's 2.5 kilometers away, so we would finish this dive, but we are ahead of our time.
Yeah.
Because there wasn't so much to see, so.
Yeah. That's a 180 on the heading. We're putting a big twist into the.
Do you think the visibility would be better flying uphill? Yeah.
It will be, but the problem is do 180 degrees, you're putting a twist into your umbilical.
Oh,
Yeah.
but we can do very
We're facing the slope here. If you look up, we turn to our right. If you look up with your camera back up,
Yeah,
tilt up.
yeah. So that's where we come from.
Well, no, well, pretty much, yeah.
Yeah.
And we turn to our right a little bit.
I mean, it doesn't look very promising to me. We might as well end the dive. I mean, we do have
we end
slurp.
We end the dive. Let's have a stop. We're just so surprised. Normally it's always the other way around.
Yeah.
You know, it just seems a complete waste of not having some. Do you want to sample?
I mean, we could scoop a bit of that, and that would be
yes.
sorted. We do
Yes.
that. Would it be possible to scoop something in this material here and put that in a box on the starboard side?
We could try it. Depending on how high up, just tilt down on your camera.
Yeah, sure.
It's quite high.
It's quite high and quite steep.
Oh yeah, it is. It is. Okay. It's always a bit.
Yeah, it gets very steep there in that patch.
It does. Oh, and look at that one. That's maybe a bit closer. This. But it is. Okay, I see. Oh yes, I see. It's steep.
Yeah, it's doable.
But could we settle here and collect stuff here?
Yeah, you could, yeah.
Okay, let's try to do that. So of course we plan our dives with sampling in the middle. Here there was not so much to sample, and therefore we finished the transect earlier than
But
expected.
It's very unusual for us because normally we stop all the time, but there's been so few communities that haven't been explored by the team also before us.
But it's part of exploration. It's a lot of unknowns, and you have to adapt.
Yes. Yeah, no.
But so we're going to settle here. I saw there was a little
Wherever
place.
the rough can settle,
and
then
we
can
take 2 things and then we're off. Yeah, we've been keeping going down like the last 100 meters.
I know.
Okay. It's always a bit tricky. We have to leave space for the boxes to open, the arm to move. It's very tricky, especially on a steep slope. So we'll see what can be done. Zoom out as much as possible. Are you interested in this? I don't think we've collected that species yet.
No,
Maybe for solvent? It's a
We
nice
can
color,
try.
right?
We can try. Is it slow?
Oh, it's gonna be, no, because it's gonna be, does it produce mucus? Because I mean,
so.
it could be all alone there. It's going to be really good.
Yeah.
But it's one of those. I think we've already had one.
Not in purple.
Not in purple. So, yeah.
That's the only thing to see.
Okay, we're thinking of sampling the sea star, the purple sea star. Yes.
Can we take it with a shovel?
Yeah.
And then we get some of this mat with it.
Yeah, and then I think this is Lysodendrorix. So maybe first I can zoom on it.
Let's zoom a little bit.
Yeah.
And just a sec. Can we not stop? Can we now have the lasers off, please?
Off.
Yeah, perfect. Because then I can collect this for Sergi.
And I want to collect.
And you want to collect this?
Of course,
No,
that's why I'm not zooming in it.
No, but then we can put
That
a
makes
few
them-
more things in the box.
I think we'll, yeah.
Yeah? Let's make a little close-up before we go there.
Mm. Yes, can we take the arm?
The purple one, is it?
Well,
Yes, the purple one. Yes.
the purple one and a bit of this one. So it can it all go in one box?
Yeah.
Like in one scoop, or we try several?
Oh no, you have to do two different scoops.
Yeah,
Two different
that's
scoops.
fine.
Okay.
That's in the same.
So that will be cast,
Or is it already?
probably cast 15.
I'll bring it in.
Should we fill in the...
For sure.
Went faster than I thought.
Yeah.
Well, we only went downhill.
We'll get that now, won't you?
We will sample that one now. We will sample this one with the sponge and maybe that together. So because we
Could you bring your camera out?
Yes, absolutely.
and can we nothing. There's no nothing. There was. It doesn't matter. We're done. But I think it's better to stop.
I will let you know.
Thank you.
So we're using the scoop to try to collect a few animals here. We're at 1,475 meters depth.
Way off the boulder.
76 degrees north.
Yes. The sea star is still there.
Yep.
Sorry, my eye off the boulder, sorry.
It's probably actually sitting on the sponge, so it's probably a bit soft behind it, just to let you know. Yeah, I think. Yeah, it's in the box. Yeah, I saw it going down.